Yeah, I usually do it whenever we get in the water, but it's fine if you did that.
Yeah.
Yeah.
No, I haven't dropped it yet.
Okay. Yeah, you're good.
Pilot, we'll wait for your confirmation of thrust before releasing the vehicle.
Roger that, dive suit. Pilot has control.
Pull the pin. The lift line is clear. Let's go ahead and lock in auto head in versus ship and push center line.
Roger that.
That's a good heading there if you want to just push out.
Pushing out. Coming ahead full. Heading locked in, inverse of ship.
You copy that, pilot. Other than that, let's go off stick.
Off stick.
Looking good.
Auto heading in.
All right, deck. Let's go ahead and launch Sirius. Pilot, please provide some halfway as Sirius comes up and over the side.
Backing 30%. Backing 40.
Good copy, pilot. If you get up over 60%, pilot, let me know.
Roger that. Backing about 60 right now, dive suit.
Good copy. Pilot, we're easing forward way.
Copy.
Midwest.
Backing 50.
It's looking good, pilot. Let's maintain that.
Roger, maintaining. Easing.
Okay, pilot off stick.
Pilot off stick.
Winch, 0 reps.
0.
Pilot, 0 turns.
Turn zeroed.
Okay, pilot. Target lines are clear. Let's go ahead and descend down to 15 meters. 15 meters per minute. We'll call out every 10.
Roger that, dive suit. Descending. 50.
Descending.
Ready for monitors?
Ready for monitors.
Your auto heading's still engaged for reference.
Thank you.
Rocks up.
10 meters. Wire out.
D2, 17, slowing.
Just make out your oxide.
20 meters. Wire out.
D2, 23 meters depth.
Three 0 meters. Wire out.
D2, 32 meters.
Four 0 meters. Wire out.
D2, 41 meters.
Dive soup nav, good tracking.
Good copy, nav. Five 0 meters. Wire out.
D2 holding, 52 meters depth.
Good copy. Winch, dive super.
Go ahead.
Can we get control pass to the control room at this time, please?
Control room has control.
Control room has good winch control.
Good copy. Pilot, dive super.
Go dive suit.
Control room's ready to continue with the dive. Let's go ahead and descend.
Roger that. Proceeding with dive.
Nav, dive super.
Dive super nav.
Would you suggest that we come into hold position back here on the AFCON?
I would come to an easy stop when able.
Good copy, will do. Nav, dive super.
Go ahead, dive soup.
We're in the process of coming to an easy stop at this time. Please watch streaming as we do so.
Good copy. Watching streaming.
Nav, dive super.
Dive soup nav. Go ahead.
Just so that you're aware, we are in full auto position. We are in the process of passing control to the bridge at this time. If you are ready, I'll pass comms off to you, and you can work with the bridge to make our way to the bottom.
That's a good copy. If you're ready, I will take comms.
Good copy, Nav. Navigator has comms.
Navigator has comms.
Pretty good launch for a new guy.
Not too shabby. Good job, dive soup.
Nav, dive soup.
Dive soup nav.
Bridge has good control of the ship at this time.
Good copy. Bridge has good control. Thank you.
Winch looks good. Coming up.
Thank you, Winch.
Go ahead, Nav.
Yeah, Bridge. Looks like we're holding position nicely. The ROVs are descending at this moment. We're just going to stay right where you are. This is perfect. Since we're not making any position moves, unless you need to change head, I'd suggest we go ahead and engage port prop and move rotation to A-frame.
All right, I have engaged port prop and port rudder, and I'll change the center of rotation to A-frame.
That's a good copy, Bridge. Thank you very much.
Thank you. Yes, go ahead.
We'd like to make one adjustment to the ship position before we land. Can you please make a position move 030 meters at 035 degrees, slow speed?
Oh wait, just to confirm, 30 meters with the bearing of 035 at maybe 0.2, 0.1 knots.
That is perfect. That's a good copy, Bridge.
And sorry, one more thing. We are center of rotation is currently A-frame. You're still good with that for a position move?
A-frame, center of rotation is fine.
All right.
Thank you.
Thanks.
Thank you. Yes, go ahead. Yes, go ahead.
Okay, if you can look at high pack, you'll notice ahead of the ship to the southwest, we have a very steep wall-like feature. There will probably be segments of vertical face and other segments of softer slope, but generally very steep to the southwest. With that in mind, and given the wind and current, we're going to want the bailout basically dead astern. So let's say a direction of 035 astern.
035 for the bailout. Thank you.
Thank you, Bridge.
ROV nav bridge. Just wanted to let you know that the move is complete.
Okay, good copy. Move complete. Currently, we are 30 meters off bottom, and the pilots are beginning to acquire bottom here momentarily. We're looking good. We're holding station.
Sounds good, thank you.
Video survey 1. I think we're ready for the pre-dive brief whenever the pilots are ready for the general announcement.
This is Okeanos video engineer Roland Bryan making a general announcement to all those on the ship and the shore. This is a reminder that all audio associated with this dive will be recorded with our archive video, as well as being streamed ashore via the satellite and the internet. Keep that in mind as we proceed with the dive today, and enjoy the dive. Thank you.
All right, thank you, Roland. So good morning, everybody, and happy Labor Day to those who are not laboring. We hope to bring you something fun to watch during your hopeful time off. So last night we transited away from our 10th dive site, which was in Beam Canal, and we're working our way back out to open ocean. Not quite yet. Let's zoom on over. So while we're waiting for the weather to dissipate a little bit more in the Gulf, we've pulled into Cordova Bay, which is a little bit more exposed, but still offering protection from the swells and the weather that we're seeing out kind of more in the open ocean. And it should position us to get out tomorrow and begin diving in a little bit deeper and maybe a little bit less physically stunning places than these fjords that we've been spending the past couple days. We're really excited about today's dive here. It looks like it's going to be an interesting feature. Potentially, we should see a couple of different areas where different communities could be thriving, and hopefully some diverse geology as we land at about 450 meters or 480 meters, and then we're planning on ascending up to about 320 meters. And along the way, there'll be a couple of different steps within the bathymetry that we collected last night, which is really beautiful. We're looking at a 5-meter resolution with no vertical exaggeration. So this is what it actually looks like down there. So another very impressive feature that we'll be diving at. And with that, I would like to pass it over to our science leads to introduce themselves. Jamie?
All right. Thanks, Sam. My name is Jamie Conrad. I'm a geologist with the U.S. Geological Survey. I work at the Pacific Coastal and Marine Science Center in Santa Cruz, California. And yeah, I'm looking forward to today's dive. We're diving just offshore of Long Island, which is one of the 1,100 or so islands that make up the Alexander Archipelago of Southeast Alaska. As Sam mentioned, we're still up on, geologically, the North American continent. We're about 40 miles or so, I believe, from the margin. And we're in, hope to see some rocks. I expect we will see some rocks. And these are a little bit better understood and more well known than the rocks we've dove on the last couple days. These are rocks known as part of the Alexander terrain, and these terrains, this is one of the many terrains that make up most of the west coast of North America and virtually all of Alaska. These are more or less, if not internally coherent, but internally related packages of rocks, bits of crust and perhaps some oceanic material that's been assembled or accreted up against North America by the northern movement of the Pacific Plate. In general, the process that happens is that the oceanic basement of basalt, which is created at the spreading ridges, does tend to subduct along these a convergent margin. But the things that accumulate on top of it, including the basin sediments, as well as anything else that's built on them, including volcanoes or bits of crust, tend to get scraped off and become part of the continent. And this is one of the primary ways in which new continental crust is made. These rocks are fairly old that we expect to see today. Ordovician and Silurian in age, which is between 500 and 400 million years old or so. They've from the onshore mapping, I'm expecting that we will see some volcanic rocks, and hopefully not too metamorphosed, so we might be able to pick out some flow features and whatnot. This terrain has actually been described as an island arc or rocks related to, say, those that formed on this along the Sierra Nevada of California, but probably formed from somewhere much farther away. A modern analog for these rocks that we're going to see today might be the modern-day Aleutian arc. So it'll be interesting to see what we see. But with that, I'll pass it on to our biology lead.
Hello, I'm Merlin Best. I am the biology lead on this expedition. I work for for Fisheries and Oceans Canada out of the Institute of Ocean Sciences in Sydney, BC, on Vancouver Island. And yeah, this area that we've just landed in, I see, is the exact area that this coral species distribution model I've been talking about for the past few days has highlighted. So just to give you a bit of a background, it's kind of a series of calculations that took into account 20 years of bycatch records for coral and added in all these environmental factors like temperature, depth, and slope, and produced this map that gives us a gradient of probability where you could find corals in abundance, and it covers the whole Gulf of Alaska. The ones that I have access to have the highest numbers across the whole Gulf, pretty much right here in this area, Cordova Bay. So since we're, kind of taking cover from that weather pattern that's moved through the Gulf, it's a really great opportunity to check this out. Whether or not we find a lot of corals, we're looking for those big primnoids, Primnoa pacifica specifically. But whether or not we find them, it could help us validate or ground truth that model, which is what we call it when we go to check out an area and see if it lines up with what the model predicted. So yeah, and I see right now we've landed on kind of the soft sediment base of the feature that we're going to be kind of climbing up in steps today. And yeah, we've seen a lot of this, just the water column here is full of marine snow, lots of turbidity and debris from biological activity, and a different kind of fish right now than what we've seen so far. I'm lucky to be joined by a team of shore-based scientists that I have access to via a chat room, so hopefully there's some fish biologists on board that can help us with identifications, especially when we see new things. So all of this beautiful marine snow that we're seeing is kind of the product of biological activity higher up the water column or further away that's been carried here by the current in this case. And it will all eventually settle. Some of it's recycled in the water column, but most of it will settle eventually on the sea floor and become habitat and become food for all of the different animals that live here. I think already I'm seeing some sea stars, maybe a little tiny bit of coral and some anemones, so it's already lots to look at. I think there's a flatfish down there. We had a nice zoom in of this earlier, but you can tell by those little white dots around the edges of the fish, it's probably a Dover sole.
And nice to see that the current is a little bit down from what we saw yesterday.
Yeah, it seems a little more calm. Things don't look like they're about to get blown off the side of a rock.
Yeah, a blizzard of marine snow.
It reminded me of driving in a blizzard, exactly. Luckily, this D2's got four-wheel drive. Four thruster drive. Actually, there's probably, what, like 8 on there? I have a suggestion from the, oh, a little squid, cute. A suggestion from the science chat that those long, thin fish could possibly be herring. So if we see any more, once we get calibrated, we'll take a zoom and see if we can confirm. Curious little herring, if that's what it is.
If you're watching at home, I believe on cam 3, if it's not there now, it may be there soon, is a 3D rendering of this face we're going to be climbing up. And right now you can see in real time a model of D2 down in the lower right corner, and you can see the up above us the orange-looking cliffier, possibly outcrops, but anyway, terrain we will be climbing today. If we zoom out a little bit, we can see this package is perhaps bounded by maybe a fault on either side that's juxtaposed this group of rocks in front of us from separate blocks to either side. And those little canyons or valleys that kind of are forming a little V shape now in the view.
And that image on stream 3 that we're looking at is colorized by slope. So that's something that we like to look at when we're picking a dive site for a number of reasons. One, in some of these high slope areas, you have a better opportunity to see exposed rock, so better attachment points for a lot of the organisms we generally look for, and exposed rock for our geologists to look for as well as perhaps more importantly, is it gives us a little bit more information about the safety of the area as it pertains to piloting the ROV. So for a number of reasons, we like to have areas where we can look kind of dead on and go upslope with the contours, so we're not running into any surprises, and we know that we're always kind of looking up. And that's good for safety of the piloting of the vehicle, as well as for the quality of the imagery that we're collecting. Whenever we're looking down or going down a slope, you'll see a lot of blue water. You won't quite be able to see the bottom as well as when you're looking upslope. So we generally look at the slope maps to both tell us what directions are best for our view, as well as for the safety of the vehicle in general. And especially in these areas where we have high currents and potential for those currents to shift and be dynamic, we want to be able to make quick decisions if we need to bail out of an area for any reason, if we feel that it's become a little bit hazardous to the vehicle. And that way we can communicate quickly with those driving the ship to help with some of those moves and make sure that we are taking our time and being as safe as we possibly can.
And I'll point out now that what we're looking at here is from mapping that we completed just a few hours ago. As a geologist, much of what I refer to and much of what we know, almost everything about what we know about the geology of this area, has come from exposures on shore. So what we're looking at here is essentially brand new information and essentially the baseline information we need to start mapping geology, and in that sense, already we can see a little bit of new information. We're looking at a couple different packages of rocks and possibly faults that are brand new information for geologists, and ultimately will help complete those geologic maps out into watery areas where mostly we, in the old days, have tended to just color it blue and go, we don't really know what's lying underneath all this water. This is really neat to be out here.
Yeah, and then kind of similar to what Merlin was saying about ground truthing those models for coral distribution, the ability to go down and collect samples and take video of these types of features that we're seeing in the bathymetry allows us to ground truth the bathymetry. So then if we're seeing a similar type of thing in somewhere else, we have a better inference for what we might be looking at. So I think it's really important to help us better decipher. I've been looking at bathymetric models and maps for most of my career, so I feel like I know what I'm looking at, and it's good to get either confirmation or a recalibration of that as we go down and see what all those dots and pixels actually represent.
Yeah, it's pretty cool to be able to do. Do things like turbidity, like all of this stuff in the water column, does that ever affect the scanning that you do?
It depends. And a lot of work with sonars, the answer will be it depends.
Yeah. Okay.
The frequency at which our sonars operate, so our multi-beam system, which generated this, is a 26 kilohertz system. And so with all sonars, you're basically balancing resolution versus depth. So you can get a higher resolution with a higher frequency system, but you're sacrificing the depth in which it'll go down because the signal will attenuate through the water column. So with these systems, it can penetrate through this turbidity no problem. The bigger issue that we run into, especially in dynamic areas such as this, is salinity. Because basically what this data comes from, each of those points, is a measurement of time more than anything else. So the sonar sends down a signal, and then it basically just counts until that signal returns. It divides that in half, and then you get the depth. So I was always taught it in physics as the dirt equation. That's what stuck with me. So it's distance equals rate times time. And that's all that's happening here. You know the time because the sonar is measuring that, and the rate is determined by the speed of sound through water. And that's what can change. So depending on the density of the water, or basically how warm or cold it is, how much of it's stacked on top of each other, and what's in it, the salinity or the conductivity, will determine how fast that sound can travel through the water column. So that's something that we measure quite closely, especially in these dynamic regions. And we can do it with what's called a CTD, which we actually have one on the ROV, and that's kind of ubiquitous in marine science research. You're always kind of trying to figure out those oceanographic principles, and CTD stands for conductivity, temperature, and depth, and through that you can infer sound speed. But we also use, and I think you saw these yesterday, these XBTs, which are expendable bathythermographs, and that just measures temperature, and you can tease out some of those other parameters by looking at basically a database of other data that were collected by CTDs. And the nice thing about those is we don't have to stop the ship. We can do it while we're moving. So it's a really efficient way to collect that sound speed information.
Versus pausing for 6 hours or so to collect it with a CTD.
Yeah, and for the data that we're collecting at these depths, getting that CTD profile won't make the data that much better. Our uncertainties will still remain close to the same.
Oh, very cool.
But in areas like this, I spent most of my career before I came to NOAA Ocean Exploration on a different NOAA ship, the Fairweather, doing coastal ocean mapping in areas like this, including a little bit further up this strait, is where one of the first projects that I worked on was just, I think, 2 or 3 miles away from here. So it's kind of a coming home for me. But I do know probably better than most how dynamic the water can be in these areas, and especially when you're, I think the more beautiful the place was, the more annoying the work was. So it kind of balanced that way.
Oh,
so when you're working by
very
like
beautiful outside.
Yeah. But working by waterfalls really makes that a lot more difficult. You get fresh water coming in, you get the tidal changes, you get all of that. So being able to model that well with the CTD profiles was pretty critical, and definitely took a lot of work.
Yeah, this visualization is new for me, being able to have a little mini D2 kind of climbing up these features. Really helps to just put it into context in a way that I've never seen before. You can look at a map and contour lines and sort of imagine, but this visualization is pretty incredible. I don't know how I'm going to go back after this. Oh, I think that's a skate.
Oh, my.
Yeah, that's a skate. It just swam by. Watch lead here. Ooh, yes. Yeah, I think so. Thank you. Perfect. So we're just, we paused for a moment to collect some water sample here for a study of its eDNA or environmental DNA. We'll take that water, and technicians up in the lab will filter it and keep that filter for genetic analysis. The idea being that anything that swam through or the current went past in the water column will leave a little bit of its DNA behind. And we have the technology now where we can sequence parts of all of the DNA, parse it out, and get a profile of the animals that are in this area. Watch lead here. That's correct. Could we pan to the right if you're taking requests? What is that? Oh, think it's just seaweed.
A very fish-shaped seaweed.
Yeah.
After seeing that skate kind of cruise by.
Exactly. Now I've got my eyes
him.
out. Yeah. We haven't had a really good skate sighting yet. Have found a really nice skate egg attached to a sponge a few dives ago.
But yep, that looks like kelp to me.
That's great, thanks. I don't think we need a close-up. We can continue. Yeah.
And we got a question in the chat if we use ADCPs for current profiling. And yes, so an ADCP is an acoustic Doppler current profiler. So basically the same way that sound changes as a train passes by, it changes pitch, the sound that we emit from the ADCPs will change its pitch in general. And you can measure the speed of the currents through the water column with that. We generally don't run those all the time when we're mapping. One of them interferes pretty greatly with our multi-beam because they're at similar frequencies. But whenever the ROV's in the water and beforehand when we're doing our tests of the drift and making sure that we're seeing what we're seeing at the surface and what we're seeing down below either match up or is something that we can account for with the vehicle motions, we'll have those running. And we have two separate frequencies. We have a 38 and a 300 kilohertz. And the reasons that we run both of them are for the same reason I kind of started the discussion with the range resolution. So with the 300 kilohertz system, you're able to see a finer detail of the more the layers higher in the water column. And then with the 38 kilohertz, it penetrates a little bit lower and you can see deeper. And so you kind of get a full look at what the currents are doing throughout. And we have that running through the entirety of the dive so we can see how that's changing and then adjust the way we're piloting. And it looks like we got a little hermit crab making an appearance.
There he is.
A little hermit crab in some of those kind of pyramid-shaped shells, the Calliostoma probably. And I think hermit crab, I believe the family is Paguridae. And I'll probably leave it at that, but we're getting a nice close-up of this one right now. Maybe, I think there are two separate shells that may be a little snail shell of some kind underneath.
Is he thinking about changing shells here?
Could be two crabs.
Or is it two crabs?
There we go. I was like, how many big claws?
Oh, yeah, there are all
counting?
the big claws.
Yeah. Just visiting. Wow. Never been able to get quite such a good close-up of the eyes of a hermit crab. These are obviously really small organisms. The zoom level that we can achieve with this camera is always constantly blowing my mind. So you can see those two laser dots, the red dots floating in the middle are exactly 10 centimeters apart. So this is the tool that we use to kind of gain a little bit of perspective and get some detailed measurements of what we're seeing. Otherwise it can be really easy to get a little bit lost. Lots of sea stars so far. Oh, there's a big fish. Oh, that could be what we were maybe what we were seeing before. I think it could be maybe a halibut. Oh, or a sole. No.
I think it's sole.
There we go. I'm seeing those little white markings around the side. I guess that could be what we saw in the distance earlier.
Dorsal fin up and got excited.
Yeah. I mean, the sole is still exciting.
It's
Look
very
at
exciting.
it. I love how they're twisted around. So, yeah, the way that these flatfish have evolved with their two eyes on the top and then the rest of their body twisted around so that one side is flat against the ground.
And they don't start their lives that way. The eyes migrate over after their larval stages. It's
cool.
Even
I love that.
weirder than...
Even weirder. Yeah, the mechanism that must get set off to make that torsion happen from between the larval stages, that is pretty cool. See, I think these are, again, the Pacific Dover sole. Let's see.
And I imagine these mouths to the sounds are probably a really great place for a flatfish to live. You get high currents, a lot of nutrients.
Yeah, this is a pretty high abundance compared to the, kind of scattered sightings we've seen so far. So I have a couple of facts. The Pacific Dover sole, Microstomus pacificus, ranges from Baja California to the Bering Sea. Takes its name from a resemblance to the common sole of Europe, which is often called a Dover sole. So this is the Pacific Dover sole. Every now and then I have a few fish facts. We do see these from time to time on our seamount dives. Oh, there's a nice big jelly.
Maybe Solmissus?
Sure, sounds good. Oh, there's another one of those white Henricias with the red spot in the middle.
Oh, yeah.
We saw, and maybe a Chiraster, the unidentified species that we were able to collect yesterday. Maybe. Hard to tell. It's a really big brittle star. Apparently there are a couple of species of those Henricea sea stars that have the red dot right in the middle. It's very dramatic. I love it.
So here we're kind of making our way up on the soft bottom to the top of what we would refer to as a fan. That's probably getting a fair amount of sediment, seeing that we're kind of at the mouth of a valley, seafloor valley that's coming down from to south of us. So,
ooh, I think this is one of the corallimorph anemones. Just judging by those little balls at the end of the tentacles, a little bit different group from the anemones that we have been seeing. I think they're always really intricate and beautiful. You can see a couple of different size fractions of these tentacles. Really cool coloring.
And I referred to this as a soft bottom, but when we zoom in, you can see that it's actually a pretty rocky, pebbly bottom of loose pebbles and cobbles, which isn't terribly surprising, as we've seen the currents are pretty strong back in here, and likely any fine sediment is going to get swept away in these currents. So we're looking at just a little thin veneer here of mud or silt that's probably pretty ephemeral.
Whoa. Okay, this is so much weirder than what I thought it was. What are we looking at? I thought it was... Wow. Looks a little bit snake-like.
It looks very snaky.
Very snaky. Is it a snaky worm or a wormy snake? We have a ribbon worm, Nemertean. I've never seen them kind of look like their sort of head is up and searching around. Very cool. So
I think we're going to get a little closer. Yeah, wormy snake was my gut feeling, but seeing it look around like that
I know. Yeah, a snaky worm, I guess. Wow. But like even it looks like it has that little, almost like an eye spot. That's wild. So we have a suggestion from the science chat, a Nimmertian ribbon worm. And it has some really distinctive patterning, that stripe along the end. It looks like it's kind of searching through the water column for, I would assume, bits of this detritus, but it doesn't have to look very far. It's pretty much everywhere.
Yeah, it looks like it can compress and extend its body to a pretty good extent.
Oh, wow. It does feel like watching a snake in the, you know, in the grass. Very cool. I have never seen a worm that looks like this.
It's pretty large, too.
Yeah.
It was
Let's
greater
pan.
than the 10 centimeters.
Oh, was it? Oh, wow. It looks like it's injured back there. It's missing a bit of its... There's a break in the tail. Oh, no. Just buried. Just buried, I think. Wow. Yeah, me neither.
That is
amazing.
very cool.
Yeah, well, this close-up you can see, I mean, there aren't a lot of identifying features I wouldn't think that you can see from a video. I assume this has to be examined in the lab to take it down to a species, but that white stripe that runs along the whole length of the worm seems pretty distinctive to me. Very cool. No, it's not on my list, so I think we'll proceed. Thanks for that. Very cool. A really big flatfish.
I am starting to think that skate we thought we saw before was one of those.
Yeah, me too.
It almost tricked me again.
Yeah, I think we know this one, the eel pout. They make those really distinctive little circles in the water column.
That's a beautiful shot with all the
Wow.
marine snow.
Yeah, it just looks like it's drifting among stars in outer space. I love these. Oh, that's so cool.
Wow.
So I don't know. I've heard a couple of things that could be kind of resting and slowing their descent. Or they could be a little bit stunned by the lights, and that's a defensive reaction.
The eel pout version of the fetal position.
Really? Oh, good. Okay. Yes. Okay, we've had a replay, and that was a skate.
Okay.
Great. This looks like a Hymenastra sea star, Hymenaster, and a big scale worm towards the bottom. Oh, do we have a skate? Yes!
That's a
Speak
skate.
of the
Oh,
devil.
beautiful. Wow, okay. Try to pull up my references. There are a few different skate species known from this area. Looks a little bit familiar.
Mitchell Hebner suggests a big skate is Raja binoculata.
Yeah, there's also the long nose skate, which I immediately thought of because of
The long
the
nose.
kind of longer nose. Raja Rhina. We can review the video and take a good close look, but I think I saw there's two darker dots on the wings. Look at those. Look at the eyes on this one. Oh, I think they're so cool. So I think I'm seeing, I mean, the pattern on the scales is highly variable among these fish, but if I saw this in my annotations, well, no, I'm not sure. The other ones I'd probably lean towards deep sea sole, but it's giving me actually maybe more halibut. Let me see. This is what happens when you get an invertebrate biologist to check out vertebrates. I'm getting a handle on the skates, but we'll at least collect some really detailed video for others to examine later. Yeah, I'd love to zoom on this sea star. Thank you. Oh, I think another Henricea with that little red spot in the middle. Great. Some fan worms, lots of encrusting sponges. I'm seeing brachiopods, I think. Oh, no, maybe could be little scallops. A little gastropod shell to the right. Lots of empty tubes and full tubes. That kind of fan shape coming out of that tube is a worm, probably a serpulid, I think.
The way the sea star is splayed out reminds me of those old crime scene chalk drawings.
It does look very dramatic. I think either a Hippasteria or a Ceramaster.
Oh, what's this?
Something coming by.
Ooh, a really big rockfish. Wow. Okay, this could be a yellow eye. Guess I'll just have to keep all of my fish reference tabs open. Yeah, this is definitely a larger individual. So some of these rockfish have lifespans, close to humans, but also more. I think I read somewhere age estimates of closer to 200 years. But
wow.
Oh yeah, a little amphipod, I think. A little bit of damage too, maybe underneath. Looks like on this kind of cheek part under the eye. Hard to say. Ooh.
Wow, look at the detail on that eye.
Yeah, just stunning.
Levi must be piloting. He's our eye guy.
You can see the spines on, kind of decorating all different layers of this fish's head and the dorsal fins. So we'll definitely get a fish biologist to look at these and confirm, but my tentative ID is a yellow eye rockfish, definitely in the genus Sebastes, but I may be a Sebastes ruberimus. Ooh. It's looking very coral-like to me.
And the substrate is getting a little rockier all of a sudden.
I was going to say, it's a good sign when we see our first coral on our first hard substrate.
Yeah, that's a great sign, and it's a pretty big one. This is the one that I've been keeping an eye out for in this area. This is a Primnoa pacifica, commonly known as the Pacific red tree coral, even though the ones that we've been seeing are a little bit paler, kind of a light orange or even a white color. Beautiful close-up, so we can see the individual polyps, each of those little, little units sticking out off of the main stem of this coral are the polyps. Each of those kind of has the same basic structure as a individual anemone. So they've got the tentacles and then kind of a neck, but these are colonies, so they're all attached to the same skeleton. And you can see they're covered in these really hard, they're called sclerites, so like skeletal elements that are made out of calcium carbonate and add some structure and, I would guess, protection to the polyps. And you can see why we're interested in these corals as habitat-forming structures. So they, provide a lot of height off of the seafloor, as well as complexity. So you can imagine it'd be a great place for smaller fish or invertebrates to hide. And lots of more of those Henricea, I think, sea stars are on the bottom and encrusting sponges or tunicates. Very cool to see such a kind of a larger individual just right at the base in a pretty highly sedimented area. So these corals will need some area of clean rock in order to settle and start out their life. So it's very cool to see one right here. Ooh, cute little squid. We've had a couple of these identified by experts afterwards, reviewing the video. Some of those are in the genus Gonatus. Wow. Just covered a highly sedimented area, but at least clean enough to provide an attachment point for some of these little tube-shaped sponges, probably in the genus Auletta. And this kind of branching sponge down there. Yeah, zoom would be great. Ooh, very cool. I don't know this one. I think a Dima sponge of some kind, with lots of little brittle star legs wrapped around it, and kind of can barely see parts of their central discs. They're kind of entwined in this really thinly branching sponge. Lots of hydroids and I think some little branched bryozoans, but they're and a few different sponges in this view. Very cool. Thanks for that zoom. The pink brittle stars that we're seeing are all Ophiophilus in the genus Ophiophilus. No, I think we'll just document some of these bigger ones. This is great, but I don't need a zoom. That's exciting. It's always great to see some of those larger individuals. On the dive yesterday, we had quite a few sightings, but most of them I think were under 20 centimeters, so maybe some new recruitment. I see 3 of these pinkish sea urchins. Actually, a quick snap zoom of those would be nice. Beautiful. I think I remember hearing that these were all likely Strongylocentris, but I'm hoping our echinoderm experts will pipe in to confirm. There we go, Strongylocentris fragilis. Very cool. Look at all of the different kind of red and white spines on this sea urchin, kind of slowly moving around. That's great, thank you. And a little thorny head rockfish also kind of hiding down there. That's great. I think we're good. We can continue up this feature. We've had some good thorny head zooms. I think I'm pretty confident. Seeing more of these little arrowworms or chetignaths in the water column kind of flickering, show up a little more in the sediment. And there's a bit of a plume. You can really see them.
So after that little ledgy outcrop, I think it's going to flatten out a little bit, judging from the sonar, but maybe what we're starting to see is one of those kind of similar to what we expect to see as we head on up, as alternating steeper cliffy slopes and flatter areas than above them. But, well, this is pretty steep. Rock right here that's in front of us.
Wow, it's just covered. There's a lot more of this sediment maybe that's sticking around. There's a nice boot sponge, kind of a tube-shaped one. In the family Rosselidae. Hard to tell the exact species from imagery, but there are probably six or seven or more different species it could be. Oh, another flatfish. Some encrusting sponge, some of the maybe bright, whitish or yellowish. ROV, could we zoom in on that funny thing on a stalk to the left? don't really know what that is, so it's a good reason to get a zoom. Oh, wow, that's really weird. Huh.
Almost looks like a pineapple.
Yeah. So a sponge of some kind. You can see the like osculum at the top, and oh, this looks like there's something on it. A little scale worm, maybe. And it looks like there are shrimp on it, kind of hiding out between these bumps. But I'm not. Yeah, it could be a juvenile of one of the larger sponges we're seeing. But this it has a very distinct stalk. Very cool. I think we'll get nice, this really excellent zoom to kind of document some of the identifying features we're seeing, and maybe we'll keep an eye out for more. Wow, very cool. Okay, that's a new one for me. I love it. So yeah, because of its size, it could be a smaller individual of some of the larger sponges that we were seeing, yesterday, and maybe further up on this dive. So
does that lumpy look typical of younger sponges, or?
I mean usually I find the younger sponges look like mini versions of the larger ones. So it could be maybe one of these no, it wouldn't. Those little tubular shaped sponges don't end up looking like this. So no, that one's really cool. RV, could we look at that little funnel shaped sponge just below to the right? You got it. Just a snap. No, that's great. Just as we can get a focus on
that's neat.
Yeah, that's great. Thank you. Yeah, I think that looks to me like it could be a little mini Conilasma, one of the glass sponges, maybe. It's very small. Cool to see some, smaller individuals. And then lots of those little tube-shaped ones, the Owletta sponge. It's one of those spiky sea cucumbers, kind of a related species to that California sea cucumber we were talking about yesterday. And some of those little branching structures that were passing by, the white ones are hydrocorals.
There's just so much life on these rocks.
Yeah, there's a lot going on. It's nice that there's a little less sedimentation on the really almost vertical parts, so you can see a bit more about what's going on.
Here. Yeah.
Oh, a couple of big rockfish. Yeah, there's at least maybe two. A little tiny fish. Oh, those are so cute. Yeah, possibly a yellow eye rockfish. And a nice big one, too.
Yeah, this one looks a little different than the other ones we've been seeing.
Mm-hmm. We were seeing some of these kind of banded or striped ones a couple of dives ago. There's one species called the red-banded rockfish, but I think there are others that sometimes have banding like this, so we'll just say genus Sebastes for now, but there's quite a few. That one looks different too.
It does, yeah. They all...
Some of them look very different as juveniles than the adults. They have really different patterns, so it's even hard to tell smaller versions from the adult species. Beautiful. That rockfish is just kind of hanging out right at the base of one of these larger, structures. And it's just covered in a lot of different kinds of sponges. It's kind of orange and yellow encrusting sponges, definitely different species based on the patterning you can see. And lots of these, the little white branching structures are kind of pinkish that you can see are hydrocorals. They're kind of like little mini versions of the other corals. They don't seem to get as big, but they have really intricate branching, and their skeletons are like really hard, kind of solid structures. Wow. I see an anemone peeking out down there, and just, covered in brachiopods and these little tube-shaped sponges. A lot of those branching structures underneath could be hydroids, worm tubes, all sorts of stuff, just covering every inch of that rock. Bad news for the geology. Oh, look, there's a four-armed sea star, too.
Yeah, a little bit of exposure on that face. If we could zoom in a little bit on that lower part of that outcrop or face. Yeah, just heading over to the kind of lower left center. Lower right center.
I think right where that four-armed sea star is.
Possibly, yeah.
That's a good spot. Get a two
I see
for one.
I see some kind of roundish look to maybe the layering. Yeah, that's good. And a little bit to the left of here on more of the open face after everyone gets a good look at the sea star.
Mm-hmm.
Yeah, okay.
What
really
sea star?
hard to see any rock.
No, not seeing much rock. Yeah.
Wow. Hardly any.
Yeah, we can keep going.
I'm thinking... It looks like we're going to shortly traverse over a less steep area and then get to a more steep area.
Mm-hmm.
So
Right.
We're looking for rock for you, Jamie.
Yeah. Well, this is rocky.
Yeah, there certainly are lots of rocks here, but...
We're a little late to the party.
Yeah. We're good. I think keep going.
Yeah, if we'd been here 12 or 14,000 years ago, we could have seen them freshly scraped by the glaciers and get a real good view of them, but everything's taken over since then.
Snooze you lose.
Could we snap on this sponge up ahead? And also that orange thing could be a big anemone. Yeah. What is that? Like a really large anemone retracted, or a different kind of sponge?
Sea star feeding in the bottom left, too.
Oh, yeah. Oh, totally.
Mm-hmm. There's a lot going on right here.
What is this thing? Yeah, thank you. Oh, okay. I think I see an oscule, an opening, so maybe it is a sponge. Oh, no, there we go. Anemone.
Oh, wow.
Wow.
I did not expect that.
Me neither. Yeah, you see a few of the tentacles poking out, but almost the whole thing is sort of balled up and retracted. It's very cool to see this this shrimp associated with it. It looks like it's maybe, maybe just feeding on the organic matter that's attached to it.
He's here doing just what we're doing, trying to figure out what this is. And
yeah.
he just determined it's an anemone.
Also curious, yeah. So
it's
it's a huge anemone,
away.
too. Yeah, like I changed my mind. Yeah, I'd love to get an idea of what this one is. Again, some of these larger sponges are important habitat forming structures. And yeah, hard to tell what this one is. It's definitely moving around quite a bit in the current. Kind of gives me the impression that it's a bit more. Oh, someone got bingo. Maybe more of a demosponge. It doesn't have the kind of rigid skeleton that I would expect a glass sponge in that morphology to have. But take a closer look at the video. There's another boot sponge, and of course every inch of this rock face is just covered with something.
And we see all these bivalves, which are, as far as I know, exclusively been brachiopods, and not clams or mussels. Are clams or mussels things we'd see in shallower water? And this is more of a brachiopod depth? Or is it more environmental than that?
That's a good question. I don't know a lot about brachiopod distribution. Yeah, I definitely do see like different kinds of scallops, and of course the chemosynthetic clams that we know from seep areas. So every different species has their own kind of depth band, and this is not one that I've studied. I'm kind of surprised to see them in such high abundance, but here are a couple of coral skeletons. Yeah, could we have a quick zoom on the skeleton? Looks like it's covered in brittle stars. Wow. I think it's a skeleton from one of the primnoas, just judging by the size and the shape. And they kind of get, kind of a golden color that turns a little bit darker over time, I think. Very cool. So we know that there are different kinds of animals that predate on corals, sea stars, and nudibranchs. So possibly that's what happened, and then the skeleton is all that remains. Wow, this is a huge rockfish. Another one of the, with the red banding.
I have a feeling we might be seeing this rockfish's eye.
Yeah. You know what to do. So again, some of these rockfish have a lifespan similar to humans, or maybe even a bit longer. And I also, on the eye. Oh, okay. Guess it doesn't seem to be too bothered. One thing I also really like that I learned about them is that certain species like to stay in the same area. So divers have documented seeing the same individual rockfish close to the same rock years apart. So revisiting 2 to 4 years the exact same site, and they've seen the exact same individual rockfish. So they have a really high preference for kind of staying in the same area, at least a few species that are identifiable as individuals. Often it's hard to tell, but some have more distinct markings than others. There's a nice rock. The really big hydrocoral on it. And another one of these large sponges. If you could get a snap on this sponge. Thank you. Just like the tissue, those, yeah, just kind of the pattern. If we can get nice and close. Ooh, beautiful. So I'll be able to look at this. You can see definitely lines of these spicules, the skeletal structures, radiating out kind of from the center and creating almost like a little bit of a honeycomb pattern. If it's possible to see the attachment point, just a quick look. Just a quick look could really help with this ID. Oh yeah, so it's pretty different. Kind of has the same general shape of like the conylasma sponges, really large glass sponges we were seeing, but I'm thinking this might be something else. It is helpful. Still don't quite know what it is, but it's great to have this angle. Okay, that's great. Thank you.
So I think we're now kind of just up above that first cliffy looking outcrop. Now my guess is, and guessing's always a bad thing to do in geology, especially with little or no information.
Oh, I do it all the time.
But we're perhaps looking at kind of sticky little flows, and each one of these cliffs makes up one of these kind of restricted flow units that are, as we can see in the visual, perhaps several to 5 or 10 meters high, and tend to pinch out to either side, making little bodies 50 to 100 meters across, perhaps as little tongues of flow units.
Is that Chris? Did I hear something? Maybe not.
I didn't.
I thought I heard something. That was probably it.
Huh? He did pipe in with an ID of that Sea Star Saramaster. Oh, that was good.
All right. Bright orange anemone.
Mm-hmm. Yeah, I think one of the ones we were confusing for cup corals in our first dive in this area. And some really thinly branching, probably little bryozoans. See the fan down there,
right.
The kind of alternating red and white banded fan of a tube worm.
Oh, okay, yeah.
A little tiny squid, a couple of them.
That looked like it, maybe, well, I'll guess it was a rockfish, but he looked a little bit gray.
Ooh, there are a couple of rockfish and rockfish relatives that are grayish. Yeah, lots of encrusting fauna. Yeah, a quick zoom of this would be cool. I think it's covered in brittle stars and an anemone. No, yeah. Wow. See some shrimp. And I think higher up, one of those gastropods, Calliostoma. Wow. Yeah, so this was a pretty old, large individual. Could we get the lasers on the base just to get a bit of a size estimate? That's great. Yeah, covered in those little hydrocorals, I think. Most of these are in the genus Stylaster. But yeah, they're called hydrocorals because they are hydrozoans, so they're more closely related to those little branching, bushy things that we see closer to the rocks than they are to the octocorals that we've been seeing, like Primnoa.
Here's a little bit of
wow.
a forest we're coming up into.
Yeah, but really interesting, like missing all of the tissue at the bottom.
Yeah.
So something looks like it's maybe predating on it from the bottom up, and I think I see those little Calliostoma gastropods, little snails that might be the culprit. Also a lot of, I think, Hippasteria sea stars at the base of each of these.
Oh, yes.
And a big nudibranch. It's kind of a...
free for all.
Yeah, it is. Or maybe the Calliostoma are taking advantage of some space that's been cleared by something larger. It's hard to say, but there's these beautiful corals are really being munched on by all sorts of things. Yeah, so really beautiful shrimp kind of hiding. Whoa. Wow. So you can see the same kinds of polyps that we saw in the younger individual. This one's a nice deep orange color, but it looks like the rest of this tissue maybe isn't long for this world, judging by the fact that it's missing so much near the bottom. And these polyps can't fully retract. You can imagine being covered in these hard calcium carbonate scales almost, that it limits the amount that they can retract into the tissue versus something like the bubblegum corals we were seeing earlier on an earlier dive, where the tentacles and the polyps can completely retract. Wow. Look at all this.
Is that a nudibranch?
Yeah, really big Tritonia nudibranch, and a sea star. So I think this coral is being viciously attacked by a couple of things. Of course, there's some nice new recruitment growing just to the left. So I think this is a part of the natural cycle.
So how fast do these grow? It's surprising nobody's thought yet to go over and munch on that younger one.
Yeah, I would imagine that it would take, they seem very slow moving, so I think it would take a long time to go up one. This one here is just starting. The growth rate, and there's a little new growth at the bottom there, I would say. Yeah, so maybe this nudibranch either missed that part, or it's a little bit of new growth. Sometimes you do see younger corals growing at the base of much older skeletons. Wow. Yeah, this nudibranch is doing a great job of clearing the tissue, and the sea star.
And the sea star, wow.
Yeah. And the Calliostoma gastropods. So I did look into it a little bit of estimates from, I think, radiocarbon dating of, in order to do that, obviously you need the base of the coral. And so the estimates I saw kind of varied really widely. One of them said, like, the conclusion I read was under 300 years. And then I think kind of ground truthing it with other methods put it in the 100-year range for the individual coral that they looked at. Wow. Yeah.
So in between these big cliffy exposures you can see in the bathymetry and in the 3D model, if you're looking at Cam 3, are these more flatter areas that may be underlain by more thinner bedded flows that don't make these big cliffs, instead make more benches. Here's one that's been
completely
eaten.
Denuded, I think, is
denuded,
a word I see sometimes.
Denuded by nudibranchs.
There you go. Oh, a really big, boot sponge. So a couple of different species that these completely kind of clean ones, like devoid of sediment on either surface. There's a few different genera I think that they could be but they're all in the family Rosselidae, so we'll call these a Rosselid sponge.
This one has quite a bit more of the debris, water column debris, stuck just to the very lip.
Yeah, just around the lip or the outside of this osculum. It's very interesting. This sponge has some kind of mechanism to keep the tissue clear on either side, and I think it has a tuft of longer spicules, those silica-based skeletal elements of the sponge along the top that maybe the cleaning action can't reach. But then other sponges in this group only seem to keep one side clean, usually the inside in Rhabdocalyptus, so they're covered in sediment on the outside. And then another one that has sediment on both sides and doesn't seem to mind either. So they're interesting, and they can help us identify them. But of course you need to have a really good in situ image, a tissue sample, an ID, and then be able to kind of relate that back to features you're seeing. Here's another, I think, Hippasteria sea star predating on this Primnoa pacifica. Or grazing, depends on who you're cheering for.
Yeah, it's easy to pick sides, but sea stars need to eat too.
This is true. This is true. You can see a few of these polyps are kind of looking a little more limp and less active than some of the other ones. The ones in the foreground you can see are still, at least mostly extended, and these ones are, I think, as retracted as a Primnoa polyp can get. So it looks like these ones are out a little bit more, maybe possibly feeding. Where did you go? Yeah. Oh, that's wild. Very cool angle. And the little snail, you can see their little eyes.
Very interesting texture on the sea star.
Wow,
little, almost grains of rice, wild rice looking.
It does, yeah.
Texture.
Yeah. They look like they kind of are in various positions of being open or closed, which is interesting. And little kind of blunt spines in between. The pedicellariae of the sea star, perhaps. This is a really beautiful, vibrant orange color on this Primnoa. I think I can see little eyes from some shrimp hiding out in there.
Hey, this is Chris Ma.
Oh, hello.
Hey, you have some really beautiful video here, and even though we've seen similar types of predation in like the Hawaiian area, I don't know that I've ever actually seen them. I've read about these in the literature. I think the paper by Krieger and Wing used to document these as sort of SPA, but I've never actually seen them myself. And so this has been all very sort of really wonderful video footage. The things that you were looking at earlier, all of the texture, and this is something that keeps on coming up that I haven't had an opportunity to really discuss at length, But when you guys are focusing on the surface and you see all of those, for lack of a better word, the things that look like little lips or little eyeballs or whatever, those are called pedicelaria. And so the surface of these sea stars is covered with them, and it's not entirely clear what they do because they're on the bottom surface as well.
Okay.
And when we saw them in Hawaii, they would open and shut. I mean, they looked like little jaws, and so I'm not entirely clear. It's not entirely clear to me why they do that. Why? So I mean, at first I thought maybe there's some kind of battle going on with the sea star and the stinging cells on the coral. But obviously there's a whole bunch of these pedicelaria that are facing away from the animal, which would imply that maybe they're used as a defense against predators on the surface. But there aren't. When we see them, there aren't any little like crustaceans or anything around on the surface. So, it's a big mystery to me. I don't know what they're for. I mean, that's why I always relish a close-up look, because hopefully one of these will provide some kind of insight, as to, what is possibly happening. Because, I mean, it's occurred to me, for example, the possibility that, so one of the things that I have seen in other video is that those soelaster that we see so frequently will, while these, will just roll over Hippasteria and just eat them. They have this notoriously complicated behavioral thing where they're that was called in 1967, I think. There's a paper that a whole bunch of jolly marine biologists called the 13-arm hammerlock, and the soelaster sea stars do this to other sea stars where they just kind of wrestle it down and then proceed to eat it, which sounds kind of funny, but is pretty horrifying, I would imagine.
Yes,
but I've seen video of, or at least imagery of soelaster feeding on Hippasteria. So maybe they're literally they've just evolved this, this defensive structure to prevent other sea stars from getting on top of them. I don't know, because we don't have any. I've never seen the Hawaiian ones with soelaster present, and Hippasteria is widely occurring. So, I don't know if there's just sort of a leftover thing, if different species have more of a, have more of a reaction, and this is just a carryover. But every time we see them, I get excited, and certainly, like, we've been seeing a lot of these, and every time we get a close-up, there's excellent documentation of these structures. So keep up the good work. And Roland and all of the GFOE people, please, whenever we see these feedings, I mean, I know this would be boring for everyone else, but nothing would make me happier but to watch more Hippasteria eating coral all day. So, there we are. But I see here more Primnoa with the post-Hippasteria handiwork. Anyway, I'll let you go. Thanks for giving me a couple of moments to explain my current love affair with goniastrids. I'll catch up with you guys later. Bye-bye.
That's great. Chris. Yeah, thank you for that.
Sure.
It's always really fascinating. Especially, I love hearing what is stumping other scientists in their field. So now I am motivated to zoom in on Hippasteria too. So we can kind of try to imagine what the function of those features that we can see might be. That's great. I'm good to kind of continue. I think we've seen a few of these Primnoa with a bit of Hippasteria-related damage. Cool to get an idea of general counts as we go along, and maybe someone in the future will measure the level of damage to each one. I know that in our annotations we always note when there's a damaged or a dead coral. It's important information for us. And a follow-up from Chris Ma, most of these look like Hippasteria heathy, or heathi, that's the species of Hippasteria that we're looking at. You know, we've seen a few different species of them so far on this survey. It's a little Ceramaster who just passed over. There's another one, a fairly healthy looking coral with a big Hippasteria sticking to it. I think some of these other sea stars could be with the really strong, I don't know, kind of outline, to me look like Ceramaster or cookie stars. I think that's that sculpin, maybe?
Mm-hmm.
There we go. I really like these little fish. There are a couple of ones. The species we were given earlier is the dark fin sculpin. Could be related to that one. And a couple of small sponges that a lot of them are demosponges, the tubular-shaped ones, probably in the genus Auletta. But a couple that look like they could be juvenile glass sponges. So we saw a couple of the reef-forming cloud sponges yesterday. So we know that they're in the area, and those could be juveniles. Wow, look at all when you look up there, the water column's just full of these little chaetognaths everywhere.
Oh yeah, they love the lights.
I think they do, yeah. A couple little shrimp swimming by, but lots and lots of chaetognaths or arrowworms. So it looks like we're approaching that kind of vertical feature.
Right, a little steeper part.
Mm-hmm.
Perhaps a thicker flow. Now, in identifying these as volcanic rocks, remember that I'm extrapolating from exposures on shore where volcanic rocks were mapped, but those are maybe a couple kilometers away. So, but these do kind of have the look of kind of tabular volcanic flow units more than sedimentary rocks. Again, these are Paleozoic aged rocks, probably of Ordovician and Silurian age, or about 400 to 500 million years old, representing kind of a volcanic arc terrain, maybe something like the modern day Aleutian chain. So we can expect a lot of variation, not just volcanic rocks. We could even be having little intervals of sedimentary rocks that were laid down in between the flow units. It's hard to tell from these exposures with all the cover, but we'll certainly try to get a piece of something loose when we come across something that looks appropriate. Yeah, maybe even one of those.
It's always so hard to tell.
Yeah, it is.
You know, in the past.
Well, they might be a little bit on the bay. That bigger one is, yeah. Nah, let's keep looking. Although I don't know if we'll see things much better, but we certainly have plenty of exposures in front of us. So.
Ooh, beautiful red squid. Wow. A little bit of ink. Cool. So a lot of those squid have been in the genus Gonatus, but we'll definitely have to have our cephalopod experts review.
Well, that might not be a bad rock. Well, it's probably a little bit big. And maybe attached. Yeah, that's too big. I'm attracted to ones where I can actually see part of it.
Smaller one behind it, maybe? To the right? Well, hard to say.
Yeah. Oh, those are some loose ones. Maybe, yeah, any of those would be good. Kind of right in the center there, just below the lasers. Oh, and a little sculpin come over to see what we're looking at. I think some of those could be picked up and are loose.
This is great. I'm also very interested in a rock sample from around here. There's so much to document and look at. This would be good for both of us.
So ROV, maybe, yeah, that one just to the right of the laser is grabbable, but any of those that look good to you? When you see just a single loose rock, there's always the possibility out here that it's a drop stone from that's possibly been dropped from an iceberg as the ice sheet broke up, and any sediment, boulders, sand, gravel, whatnot that it had picked up, then that gets dropped as it melts.
All right, we
but
have
this,
wiggle.
But this looks like when you see more than one rock in a place, you suspect they're probably derived.
Can I do a rock box, starboard rock box for this? Does that work?
Yeah.
All right.
Should try to look at it close.
You want to get extra lights on?
Lights
Zoom in?
are on at 30.
Okay. Is this a good rock?
That is that we think so.
All right.
How can you tell?
It's hard. Has things attached to it, so that'll take a while to clean
It
off.
sure will.
Let's do starboard.
Copy.
Pilot, can you hear
I'm
video?
excited about
Yeah,
it. We
I can
haven't
hear.
had a good
Can you
rock
do a
in a couple
rotate
dives.
There we go.
I'm seeing,
Let me know
I
when
think
you're
I
ready
see
for,
little skeleton
box
shrimp, those little caprellids. We'll see if they stick around for the ascent.
Can you tilt? There's the bottom of the rock. It's the
Do you think
rock.
it would fit in the bio box?
There we go. Yeah.
Yeah, I could fit in the vial.
Maybe, if you're okay with that.
No, that's fine.
Yeah, then we can kind of keep a hold of
I use it on video.
some of
Go for
the
scale.
encrusting fauna that's there.
Okay.
There's a little solitary hydroid in the
Use it on
front.
video.
I think I saw one of the
Yeah, we're gonna
chitons
push
that we were
the
looking
on
for
the
as
bottom.
well.
Lights are clear.
Mr. Fernando, you ready for drawer out? Wait, my craft's probably in the way of the drawer, so just give me 1 second.
Drawer out?
Yeah, 1 second.
Oh, it's up by you?
Okay.
Okay.
I'm going to go for bio box if you,
Oh, hold on then.
if you want to open up both sides. Alright, you can open the drawer.
Okay, hold on.
How's that ship moved in?
And of course, the
We're going up and
two
up. Where's the door?
trachiopods
Starboard, right?
attached to it, potentially in the genus
Pushing door out.
Laqueus.
Roger.
Thanks for that identification
Okay.
from Chris Ma. We've been seeing brachiopods everywhere, and not something that I've ever seen in great amounts, certainly not in the abundances that we have here. So we have a few references for identification. One of the scientists said we have collected a zoo.
Oh my! Already.
And that's not far off.
Starboard
I wonder
outboard.
how many different phyla are in there. Okay.
ID EX2306-D11-02G in the starboard outboard bio box.
You got a waypoint?
Copy 02G.
Alright, here we go.
Is that outboard or inboard?
Starboard,
Outboard.
outboard. I did say inboard at one point. Roll. Okay, here we go. We're taking off. Got more tether. I'm going up this steep section here.
Oh, wow.
Ship is stopped.
All right. Well, my day is done. We've got a rock.
Ah,
Coming up.
Okay,
flatfish to the right.
Rock. It's about.
I mean, I'm also pretty happy. There's lots of biology on that rock. It'll keep us busy for quite some time. think it's your first rock in 3 dives, isn't it?
That is,
wow.
yes.
Well, I hope it's a good rock.
Looks good to me. There may be some small Primnoa branching at the bottom of this feature.
I'm gonna snap it.
Oh, thanks, yeah, actually. Great, yeah, confirm, that's a Primnoa. Thank you.
All right. Onward and upward. Team. Here we go.
Ooh, can we zoom in on those corals right above the lasers? I think we have a cup coral, a big one. Yes, I think that's Desmophyllum.
Big cup corals. Those are big. All right, let me get
And if they are, I'd like to collect one if that's possible.
Do you think it's a suction?
No, they're hard corals. We might have to break one off at the stem.
Understood.
Because I don't think we can collect that rock. But let's get a good zoom in on them first.
Understood.
Thank you.
All right, Vinny, come in.
Wow, so somehow in the midst of all of this sediment and debris that's flowing through the marine snow, these cup corals have found an attachment point. These are Desmophyllum dianthus. They're related to Lophelia, the reforming cold water coral. And you can see the skeleton of this is a hard calcium carbonate skeleton with all these different little divisions, these little plate-like structures, and then you can see it's covered in a thin layer of tissue and little tentacles coming out. So these are scleractinians, a stony coral.
Oh, neat.
And they are big, really big for a cup coral. Some of the largest that I've seen.
So I'm thinking, take the arm, grab them, bring them in front of the suction nozzle. And
the deep sea surveys that I've been on.
Watch lead, pilot?
Yeah, watch lead.
Okay, if I grab them with the manipulator, can I present it to the suction nozzle and then suck it up? Or are these stiff?
They're very hard, stony corals. So I think you might have to try to use like one of the claws to snap it at the base, if that's possible, and then maybe feather it or use the suction to drop it in. I don't think it'll go. I doubt it will go in the suction.
Let's get lasers on.
They're out there.
I mean, I don't have to do much, just enough for you to open a box.
And hopefully there's a place for it to land on if it breaks off and not tumble down the cliff face.
Good point. We are very steep here.
Okay, so we're ready to change the monitors.
Just put your toes up against the wall. Craft it.
My starboard toes up.
Yeah, you got it. Once you come off the stow, I can take over.
Okay.
So we have a good reason for sampling these. There's, these are really rare to be able to collect. And there's some important questions, genetic questions that we have about how these corals are related to each other and other corals in, in the same group or the same family or genus. And so we'll be taking tissue samples of these for DNA analysis that could help us answer some really important questions.
I think I might grab it outside the cutters and then twist and break so it's squished. You want to zoom in on both cameras, video?
I don't know about the cutters. The hose might give you better grip, though.
I don't think that'll cut.
Okay.
I think it'll have to be a snap, and if we could... I know it's a hard ask to try to get one and not damage the other, but let's try. Wow. That is big.
I'm going to try to get it right here. Good.
As close to the base, I think, as you can.
Oh, I could get the one down there.
Yeah, could we maybe either try the feathering or the suction sampler to pick it up?
Yeah.
Yeah.
That was, look how stiff that was.
You should save
Oh,
what's in
you
your
could
claw
keep that
already.
actually as a,
I think I'll just go for the one that's on the ground here, that one.
Where are you going?
I think I might try to suction sample it out.
Pull by both cameras.
Or I could. It's pretty stiff. I could cradle it and bring it to the suction end effector too.
Okay.
Yeah, the suction might be the best to just preserve the skeleton as best as you can.
Okay. I'm gonna get the nozzle. So here.
Okay. I can pour out. Let me know.
Yeah, hold on one second.
Copy.
I'm going to switch camera 12 to jar. You want to get jar 1 ready?
Yeah, it's ready.
You ready?
All right. Bobby's flying.
Coming out. Dry lock off.
All right. Drawer coming out slowly.
Backing off. In auto depth. Holding here.
Great.
All right.
All right.
Drawers out. Okay. Let me know. You
Drawer.
can look at that camera as well for
Drawer.
reference.
Yeah, you can draw it.
Okay, pushing you back.
Okay. All right. Suction. Suction. Where do you want to start? 500 or higher?
Okay, set your toe down.
Jar light on.
What was that video?
Jar light.
Jar light.
Yeah, you can see where my toe was.
Yep.
Jar light.
Gonna put it right back.
Suction.
Yeah, you can go like 500 or 600.
Copy. Right there.
Yeah,
That's 500.
Yeah, it's 500. Copy. All right. You want to zoom in on how many these and get it a little more lined up?
Sorry. Maybe Sue's right there.
There we go. You're gonna give me 1,000.
Copy.
Give me a little tilt down.
That's 1,000.
Sorry. You want your box back?
Alright, I gotta get that. No, you keep flying. Alright, here we go.
Do you want this view over there?
No, you're
The
good.
main one?
You're good.
Okay.
I just need to get the. There we go. Are you inhaling?
Yeah. 1,000 right now.
Great. Stop.
Can we maybe wiggle it over the
Okay,
bio box and try to drop it?
Yeah.
All right. Hold on. Let's come out on HD2.
Pour it in, we're gonna blow it in.
Yep. I'm gonna pull off.
Maybe you want to hold a small suction.
I think it's in there. I think it's gonna be wedged. I'm actually concerned that it'll not come out of anything.
Okay. Port inner.
Oh, I see it.
Yeah.
Okay. Hold on.
We're gonna try to blow it in a bio box here without breaking the hose
All
out.
right. Coming out with the drawer.
What am I doing?
Are you clear from everything? Okay. Pushing out.
Hold that. Hose. Bring it around.
Great view from the aux cam.
Yeah, for sure. How am I looking, Bobby? Gotta turn a little bit. Just right. Left. Just right. There it is. Is that going in the right place?
Yeah, yep. Just had to start slow and ramp up
Okay.
slowly.
Let me know.
Hold that there, Levi.
Blow it in, Mr. Fernando.
Okay. That's
Give it a pulse.
500 to start.
Giving a little up. Giving a
a
little
little
up.
more.
Giving a little up.
There you
Hold
go.
it up.
All right.
Very
Drawer's
nice.
stowing. Oh, sorry.
Keep the drawer out so I can stow this.
Ready to see port.
Port inboard.
Inboard, thank you.
Okay.
Maybe connectivity steady. And
Sorry, I'm secure.
Okay.
oh, actually, I know this.
Jar 1 has sediment in it.
You're floating.
That's sample ID EX2306-D11-03B.
Sure.
And the port inboard bio box.
Bobby Moore with the assist.
I
Desmophyllum dianthus.
That's definitely my favorite sample.
don't know if you need to clean it out. You just need to note that it was used and maybe not use it.
He can probably blow it out. Let's see what he's got. We'll try.
Oh yeah, it's got a lot of sediment.
Watch this. We're going to try to clean out jar 1. There was some sediment in it. We're just blowing it out so we can reuse it. But if you
Can we
prefer us not
leave
to-
it in there for now, and then if we need the jar, we'll do that? But
I think
there
it might
could
already
be.
It might have already. Most of it's already kind of blown out when we blew out the.
Oh, okay. Just there might be something interesting in there.
It's mostly cleaned out now. I think. I'm just looking at the camera.
Yeah.
Can we zoom on that orangey
Yeah,
encrusting
you come in
sponge
here.
while we're here? Oh, cool.
Interesting.
Yeah, really different
Sorry, video. Floating here.
kind of
There you go.
morphology from some of the other sponges.
Don't
I hope
zoom
we
in
get a
anymore.
piece of it on your rock, actually. You never know what we're going to find on that.
Is that a good enough look?
That's
Watch
great,
it.
thank
All
you.
right, we'll proceed.
Yeah.
Alright, I'm back with you, pilot. Delta 16. Rotator at 52. Lights are still 50% video. Gonna kill him.
Seeing the dorsal fins of a rockfish kind of hiding in between these crevices. And lots of these little tiny
tether, as far as I
hydrocorals.
can tell, it's looking good.
Copy.
Could we get a snap of that yellow foldy encrusting sponge
Right
there?
here. Yeah, we snap in video. My toes up against the wall. Come in. Yep.
Cool. Wow, again, a really different morphology from the encrusting sponge that we just saw. These are really hard to sample aside from grabbing the rock that it's growing on. So we'll collect this this close-up for reference, and I think we're good. Thank you.
Okay.
There's a rock under there. Where'd that come from?
Oh, and another different species of cup coral right beside it. The Caryophilia, I think.
All right. Proceeding up, up, up. Just exploring here. Wow. Straight up and down here. Thanks for helping me, Bobby. That was fun.
Yeah, that was good.
Would you prefer flying or crafting?
I don't know.
Is it easy doing, like, manipulating the craft at an angle from the TV? I don't know how.
That's true.
It makes sense to keep with, like, the pilot is flying mentality, but there's not really much flying happening. And like Fernando said, I think you have the better awareness for the craft.
Yeah.
Especially since all I was doing was holding position, you know, off the wall for the drawer, etc. There was no real fancy flying.
Wow. I think another cookie star and another vicious coral predator, the Hipposteria. maybe taking a bit of a break. Oh, there's a big, looks like some of the Solasters. So the larger sea stars that predate on
lasers on that big
other
sea star?
sea stars like Hippasteria that Chris Ma was mentioning earlier. So now we're seeing evidence
You guys
of
like a
the
move?
reason why
Yeah,
they
you
might
10?
need to be worried.
10 meters, 215?
Yeah.
Okay.
Might get close to this this one here. Let's get up there.
Just bring the lights up at
Great, Jarvie, now.
40%.
Really impressed with
Hi,
the
can I
size of
please
this
get
one.
a move of 10 meters bearing
these two
215
here
degrees at decimal
really
2
big
knots.
examples.
Take it up another 20, please.
I'm pretty sure from the genus Solaster,
Good copy. Thank you, Bridge.
also called sun
30%.
stars.
Yep.
Lasers clear.
Wow. So yeah, definitely not seeing the same kind of pedicelaria, which is a word I just learned that we saw on some of the other sea stars that we were zooming in on. And maybe this one is a larger, sea star predator, and doesn't need them.
Yeah, I was just getting my
Although their
lights
exact function is
stay
still a
centered.
mystery. Each of the arms kind of entwined with some of those tube-shaped sponges. There's always so much to see in these zooms. I'm seeing little, tiny, tiny baby primnoas.
Oh, yeah.
Some of these white hydrocorals, probably in the genus Stylaster, that kind of lumpy encrusting
Video's
sponge.
clear.
Several other kinds of encrusting sponges, and maybe an encrusting ascidian or tunicate, I think. Of course, lots of those brittle stars.
Is that pink?
Mhm.
Same as the or is that a different
Light
hydrocoral?
Just judging by the branching pattern, I would say that it's a bit different. It's less planar than the white one,
Mhm.
kind of more bushy.
We're kind of in a forest of the white ones.
We are, yeah. It's pretty fantastic. And again, these are, really hard skeletons on these little hydrocorals, but it also makes them really brittle, so they're very difficult to sample unless you get a rock that they are growing on. I love those little guys. Ooh, this looks cool. Look at that.
This little, is that an overhang?
Oh,
A big slab?
Wow.
You want to look under the overhang?
Sure, yeah, give me some extra lights.
Let's get about 60% lights.
Looks like if you can't port, you could also see under.
Okay, apparently most of the Solasters in this area, there are many different species, so it would be hard to tell just from video. Some of them might be starfish predators, but most are predators on ophiuroids, sea cucumbers, other kinds of echinoderms.
Lasers on.
So that could be what those two were looking for. We zoomed in on earlier. This is very cool.
Looks like a couple, a slab comprised of maybe a couple flows, maybe another similar piece next to it, but these are tossed around. Loose boulders.
The eliminated section here, yeah. Hold that. It's a big rock.
Wow, yeah, so this is a nice close-up of those little hydrocorals as well. And I think some cerianthid anemones, maybe, some regular anemones, big chitin right there with a huge foot.
Lower left. Yeah.
And lots of these pink and white hydrocorals, probably in the genus Stylaster, although there are a couple of others.
Look at all this stuff.
And I think those look more like colonial tunicates to me, the white encrusting things with lots of little dots in them. Easily mistaken for a sponge, but I think they're tunicates. And of course, lots and lots of these brachiopods as well.
All right. I think we'll keep going, Ron.
Upper right. Show that
Big
section.
rockfish hanging out. Using this overhang as a bit of a shelter from the current, from the looks of it.
I'm sorry, light's clear.
So you see a pile of disorganized boulders like that. You might see something like that along a shoreline, but I think we're here too deep for this to have been a shoreline in the recent past. But we are not very far from the plate boundary and the Queen Charlotte Fault, which is a very active structure. It's in this area. It's where the Pacific Plate is moving.
Multi-beams back at 50 range.
In a right lateral sense, and a strike slip sense sideways next to the North American plate, which we're on, but
sure.
It's located, I can't remember what it looked like this morning, 50 miles or so, I think, from our location. So the Queen Charlotte, which accommodates about 55 millimeters per year of offset on it, which is a pretty good rate. Generates some large earthquakes. In the last 100 years, there have been more than 7, or there have been 7 magnitude 7 or larger earthquakes. So we're starting to get into the realm where when you see piles of rocks like we just did, you might think they possibly got jarred loose in one of those big earthquakes.
Oh. Here we're getting a nice look at that kind of the sponge I think that we saw earlier in the dive with all these little projections coming up and out, maybe almost branched in some places, covered in brittle stars. This is a demo sponge of some kind.
Yeah. Okay. Review agrees.
Very cool. I see lots of little crustaceans attached.
Yeah, you're seeing something in multi-beam. 20 meters out, 25 meters out.
Yeah. Thanks, video.
Watch leads nav.
Watch lead here.
Yes, you can see on HiPAC that we're approaching waypoint 2. I was told by the previous nav that at that point we'd kind of like to start heading towards waypoint 3, maybe along the wall or a shoulder or kind of a flat spot that we see. Is that still the case, A? And B, I imagine we'll just kind of wait for your call out to start making that move towards our left.
All right. Yeah, that sounds, I think that'll be a good plan, and we'll sort of have a chance to look at the top of this kind of a bench as we move over. Yeah.
These rocks any good?
Roger that. Then we'll continue uphill until we hear from you to start going alongside.
Sure, sounds good.
Yeah, and this might be a good spot if you want to start making your way to the south towards waypoint 3.
Roger, watch leads. I'm going to zoom in on multi-beam, which I think you can see back there. About 20 meters out, we have a feature. So we can either kind of stay on this side of the feature or...
Sorry, I don't see where you're looking.
Oh. Talking sonar.
This is on multi-beam sonar.
The orange one.
If you have that pulled up back there.
Bringing it up. Yeah, here we go. Oh, yeah. Oh, yeah.
So there's kind of the rubbly that we see now, and then this flatter spot up ahead, about 20 meters out. Would you rather we kind of wait for that or start heading south now?
Yeah, that's a nice face. We should probably go see what that is.
Roger that. So we'll continue uphill for about 20 more meters and then head southeast. Thank you.
Maybe we can call in another move, because that one finished like about a minute ago, right?
Yeah, that's right.
Okay.
Yeah, I'm still kind of far from him.
Okay, another 10.
Copy.
Bridge ROV nav. Same move, please.
Stringy branch coral center string.
Thank you.
Could we zoom in on this squirrel, please?
Absolutely. Just getting some more tether now.
Just looks a little bit different
You
from
should
what
have
we've been
tether.
seeing.
I'll come in.
I think it's a black coral.
Laser's clear.
Lasers are off now.
Oh yeah, for sure.
Hmm.
So it's kind of hard. This
Yeah, I
one's
wouldn't
really
push
caught up in the
farther than
current
this.
here. Makes it a little bit difficult to see the branching pattern, but you can see the dark black skeleton underneath of the orange colored tissue, the polyps and tentacles, those kind of strands that are dangling in the current are the tentacles for these corals. And
Co-pilot,
probably
did you
safe to
push
say it
hard
could be
after
a
this?
No, I said I
Bathypathes
wouldn't,
that's
I wouldn't push farther
kind of
than this.
caught up.
Roger.
We saw a lot of the Bathypathes alaskensis 2 dives ago.
Very different
Quite a bit of
reactions
this.
there.
And I am seeing the long single, stem coming up and then branching, so I think that's probably a good guess.
Let's look at the lowest branch.
That's great, thanks for that.
Lowest branch.
I don't think it's going to show us any additional
So these,
detail.
the polyps are kind of divided into 3, sections with 6 tentacles in total. You can see a little bit of a mouth in the middle of them. Ooh, wow.
Big
Look at that one. Whew.
Lasers on.
Go ahead.
Feels a little
Roger.
precarious.
See what's underneath that.
Wow, yeah. Oh, we'll have to take a look.
Are the
Oh,
lasers
there he is.
Yeah.
Yeah.
There now.
A couple of really big rockfish.
No, extra lights.
Yeah, bring the lights up.
Wow,
like
just
70%.
covered in those, lots more of those tube-shaped sponges, possibly Auletta. I think I'm seeing juvenile Primnoa pacifica corals, lots of hydrocorals.
We're going to keep going. We're going to keep coming up.
Yeah, a few even just some Primnoas that are just like a little stick shape
Ranging in on
with
some
polyps attached.
What was that?
Shrimp
Lights are
all
clear.
over it, and maybe some, well, definitely some brachiopods, and I think I'm seeing some ceryanthid tubes. Huh. A big sea star, I think.
All right, pilot and co-pilot kind of in that range of starting.
Seems
to
to
move
be a real
alongside.
flaky aspect to
Okay, so
the
at this point, I
flow
just
face we're
heading,
coming
and
up
I
to.
just move sideways. Is that the plan?
That's the
Or
plan
should I
if
move
you're.
like 45 on them?
Cool. A really big. Could we just take
We
a
can,
quick look
do
at
a
that big
45.
sponge?
Oh, yeah.
Lasers on.
So maybe turn to 180 and we'll
Ooh,
move along it.
wow.
I'm going to frame that up.
Look
Yeah,
at
that's
that.
the idea.
Yeah.
Co-pilot. Let me check with watch
Just a
leads,
rockfish
see
in
if they
almost
wanna come
every
up this or
crevice, and
something.
then just kind of hanging out on the vertical face of this one.
Frame up the sponge, really.
Watch leads, nav.
Yes.
So as you see, we made it to that
Lasers
feature.
clear.
The question for you is whether we kind of stay at the bottom and move alongside, or come up to the top and then move southeast. along
Lasers
it.
clear.
Yeah, let's start to move southeast now.
At the base, okay?
Yeah, and
Roger that.
get a little sense of what's down here. Yeah, before we head up.
You got it.
It's a great zoom. Thanks for that.
Yeah, co-pilot. So we're going to stay at the bottom here, and we'll start moving towards waypoint 3.
All right.
Which
Lasers back on.
from you bearing is 160, but we can do that in steps as you're comfortable.
160.
Yeah, 150 meters, 160.
right.
For pilot, it'll just be to his left, 130.
Okay.
All right. You can move going that way, right?
Yep. They want to stay at the bottom and saddle along.
Okay. All right. I think I'm stable.
You can try your heading to cut it, Fernando.
A little bit. Okay. So turning right now.
So you can just put like, see this hard return? Just put that like, you know, at your 45.
Okay, right there. That's 185. I have it there.
And then he calls
I think I'm. Yeah, I would be ready for the move.
Yeah. So like Levi just said, I'm thinking like one. I'm almost thinking like 135.
Okay.
You can go 150 if you want. That'll bring you in a little bit closer.
Either way is fine.
Whatever. Do your stress.
Really get the sense of how steep some of these are. You don't, even on the 3D perspective, you don't get that. It doesn't have the resolution to show these individual boulders and little cliffy faces.
So we'll be kind of
about
following along this this steep feature just to get a good look at it.
Yeah, that should be fine.
Maybe see.
We'll see how close I get.
See how close we can
Pilot,
get.
you okay with that?
Yeah.
135.
I'm gonna stow my starboard upper for me for that.
Copy. Your upper, your lower, both?
Starboard, upper stow.
Copy.
Well, now he's over you. He backed out, I guess.
I backed out, just so I could have room for the swing arm to swing.
Roger.
Lots more little shrimp swimming by. Of course, those little arrow worms or Keating nests kind of flickering around.
Sounds good. Okay. Stowing your upper.
Again, if you ever get a good close view of those arrow worms, or if you want to look them up to see what they look like up close, they have really vicious little teeth on them.
Yes, can I please get a ship move? 5 meters, bearing 135 degrees at decimal 2. Roger that.
So
the
these little filter feeders, the small sponges seem to really enjoy this vertical feature.
Yeah, 0.1 if that's all it ramps to. Thanks.
The sponges have little ciliated cells. They kind of have little tails on them inside of the tissue, and they'll move water and kind of slowly pump it through and filter out the little bits of detritus that they use as a source of food. Look at this. That is very cool. This kind of red banded rockfish just hanging out at the intersection between this big rock and this flatter area.
Are you going to zoom in? Or Tyler? Thanks, Fernando.
Lots and lots of sponges and little branching bryozoans. You can see, a pelagic sea star. One of those swimming sea stars. Looks like it's settling in for a pretty soft landing.
I wonder what prompted him to jump.
I bumped the wall. Maybe that did it.
So you see I think those cracks in front of us in the face are those vertical joints that
seems like I'm pretty far away from
created those slabby boulders we saw a little further down as those break loose and fall over.
Waiting for some more Taylor watch. Might grab this fish.
Smaller rockfish just wedged right in between those.
Yeah, you figured that out.
And that I wondered he did land right on the rockfish. Oh. He's going to get a ride.
Funny. What are the odds? What are the odds?
He didn't seem to wake up the rockfish.
No, a rockfish does not seem to mind one bit.
Where that puts me.
There's a couple of other things riding on this rockfish, little crustaceans of some kind.
Roger.
I think the sea star
This
is
is
upside
a
down.
beautiful close-up of the pattern of those scales. And the spines and the dorsal fin. What are the odds? That's pretty cool.
Show the sea star.
Okay.
Pilot. Go ahead.
Yeah, I think this
Roger.
sea star is in for a little more traveling than it probably normally experiences today.
Is he upside down? I think
yeah,
so.
I think I'm seeing some tube feet sticking up. So
he's trying to figure out how to get upright.
yeah. I don't think it'll take very long for this one to get flipped some kind of way.
Alright, Bobby, I got you what you want. Yeah, it looks like the pan and tilt mechanism is at 0.
Lasers on.
Pretty close to 0. Is it on video? Alright, Bobby, here you go.
Maybe turn it to port. Or we can do just 90.
Very cool gastropod, kind of a different looking shell that we got a zoom of when we were looking at that rockfish, kind of a longer one than those that we've been seeing.
Definitely more than 125. I think 150 would be good, just to get a little bit closer.
Roger.
Very cool.
Alright, proceeding.
What do you want at 150? 5 meters?
Are you okay with 10 meters?
Yeah.
Yeah, I think let's 10 meters.
Alright.
Yeah. And hopefully
10 meters, 150.
I'll be at a good distance, and then we can
It's
do
pretty
a long
impressive
range.
seeing all of the encrusting fauna. You imagine that,
Bridge, Robbie Nav.
you know, the
New ship move, please. 10
the
meters,
odds of
10.
settling on a place like a little crevice in
degrees
this rock
at
where
decimal 2.
a juvenile sponge or a hydrocoral or other kinds of corals could get enough of a toehold to grow.
Good copy. Thank you, Bridge.
It's probably pretty sparse. So there's a sea star eating something. Wow. Love a close-up if we can.
Roger.
If it's a Hippasteria, I want to see more of those pedicelaria. Oh, I think it's maybe a cookie star, though. So the Ceramaster, which I've been told eat sponges, so it could be predating on, or grazing on maybe some sponge material.
I probably want to see the other side, huh?
Yep. Lights at 30%.
Your arms are out.
What are the odds we could pan over and take a look at what it's eating? Not pan, I guess it would be a
pivot
move.
over. Yeah, we can do it in a minute. But I'm going to store my port over in a second. I gotta get away before you do that.
So you describe these things as viciously attacking whatever they're eating, even if they move imperceptibly slowly. But I wonder how often do they need to eat?
Oh, interesting. I wonder if Chris could provide us with some information. Yeah, how fast are they actually moving and eating away at that coral material? You know, things seem to move very slowly, grow very slowly in general. In you know, the time scales of things seem to be really extended.
It's proud.
Like the skate eggs that we saw, the skate egg singular that we saw the other day could take, I think, upwards of 4 years to completely, you know, finish growing inside and hatch out of that egg casing.
Wow.
Yeah, so that
video come in?
we do see things move at and grow at a really slow pace in the deeper waters.
Pretty obscured here.
Wow.
Wow, it's very precariously balanced, but it looks pretty solid.
I could buy that it's that it's wrapped around one of these little
right there.
sponges, but
Thanks, video.
Yeah, you can see its little tube feet kind of suctioning on to the surface of the rock, or balancing on those two larger arms.
Yeah, I think
that's cool. Thanks for that.
Okay, tilting back up.
Go bridge. Roger. Move complete.
Sounds good. Okay, so that's 45 degrees, Levi. I think you're at a good distance from the wall.
It's definitely slower going when we cut walls like this.
Yeah.
More tricky. You want a close up of this star watch later or not?
Yeah, that'd be great. Thank you. Yeah, this looks a little bit different from the sea stars we've been seeing.
Get a real good view of it. It's different.
Understood, really.
Color.
They're experiencing a little bit of a lag, I think, in general. Some of the folks tuning in are shore-based scientists, but
lights are
you can get some
30%.
good imagery and then maybe an identification. Yeah, the color pattern looks a bit different from also from the sea stars we've been seeing.
Tight focus.
yeah, I don't remember any pinkish ones.
No, it's actually a really beautiful color. Very cool. Some of the, kind of ball-shaped projections coming out of the dermal layer, and a really strong kind of margin. It was like marginal plates.
One more tight.
Huh.
Okay. Whatever you need, Ron.
Got a cool observation. Oh, here we go. This one looks like Geoffroaster swifty.
Okay, video.
So, or a Geoffroaster. Very cool. Thanks for that.
And enough exposed rock, you can almost get a sense of the texture. As virtually everything we've seen on this expedition, fine-grained looking dark rock. Did see a little bit of texture in it, though, sort of a horizontal-looking texture. Perhaps flow banding, perhaps, or just reflecting the flow structure within the rock.
I have an answer to some of our questions about feeding rates for those sea stars.
Fog lights clear.
So years ago, there was an observation of a Goniasterid feeding on bubblegum coral, 3 individuals.
Our estimate from
They came back a year later, and those same 3 sea stars were still there. So, as with a lot of things in the deep sea, a very slow process.
Wow.
Maybe why we saw a little bit of new coral growth underneath of the area that had been stripped away by the sea star.
What do you think, Fernando? 125, 135. Where do you want to?
Let's do. I mean,
Interesting that we saw yellow
55 from. Because you were
Geoffroaster,
right? Yeah, that latest strike was
maybe
like sea stars yesterday.
The first one was 125
compared to this pink one.
Yeah.
Just an interesting observation on the different color morphs.
Yeah, 145,
145.
Yeah, I think that should be good.
How much distance?
I think 10 meters is fine.
Roger.
So those round round spots, looking closer, they're the basal discs of some of the larger Primnoa.
15 is good.
Bridge.
There's another nice hydrocoral.
Can I please get a ship move?
Wow, look at all the little.
Meters bearing
oh, that's
145
interesting.
degrees at decimal
Can you
2.
pan up a little bit, please? That little ball with little polyps sticking out, or tentacles almost. I would have thought that was a sponge, but looking
Good copy.
closer,
Thanks,
it's oh, maybe it is just has little encrusting polyps on top of it. I'm not sure. Way more than 8 tentacles, so it looks like something else. Huh. Interesting. Thanks for that. I don't know what that is.
Maximum copy. Pretty small then.
Looks like maybe a couple of barnacles off to the left.
lasers as we come out.
Great. Wow. Yeah, every time you zoom in, you see something different. Just covered. Wow.
Video clear.
And yeah, some of those circular kind of spots that you see on the rocks, you can see a little branch coming out of them, so that tells me they're the base of a large coral like a primnoa. And yeah, just covered in these little tube-shaped sponges. They seem to really be thriving in this area.
I'm going to go for the anemone next. Watch it. You come in.
Whoa. That's an excellent close-up of this corallimorph, I think. Judging by the little white balls at the ends of these tentacles.
Frosted
a little bit different from the anemones that we've been seeing. Look at the color gradient too, from the dark purple to almost transparent is very beautiful.
And the change in texture from kind of those lines in the purple to the lumpy texture of the tentacles where they're translucent.
Yeah, it's beautiful. Oh, and there's one of those crabs at the base. I almost didn't even notice. Just kind of tucked in there at the bottom, and then another one behind. So this is often an association we see with a couple of different kinds of anemones. There's often a crab hanging out at the base. And sometimes symbiotic shrimp that also associate with these.
Fluff at the end of the stick,
I think those are, I think we had an ID from somebody in the science chat, tubularia.
Yeah, more. Is that it? Well,
that's good. Those bring up lights at 30%.
Yeah, tubularia maybe. These little solitary hydroids that seem to have a really long stalk.
It is clear.
Okay. Thanks, Ron.
There we go. Thanks to Lara for that ID. Tubularia.
Go ahead, Bridge. Roger.
Lights are clear.
Okay. I
Move complete.
Passed you a little bit, pilot. Still rotator is 58.
That's a big.
Ooh, big one.
Huh?
Wow.
Splashing.
Oh, this is also covered with something. Can we zoom in on this?
Yep.
From whatever this is.
Unless
they're really small sponges. What are they? Wow.
I'm going to get you close, Ron.
Yep. Lights at
could
40%.
be anemones also. Let's take a closer. I'll stop guessing. We'll take a closer look.
Or zoom in. What I should say? What do you want? I'm holding here.
Okay. Yeah. These. Well, yeah, look like I would say zoanthid anemones. So kind of colonial anemones. Kind of. Remind me of the stoloniferous corals we've been seeing, where they're connected by a thin layer of tissue to each other, versus the solitary anemones that we've been seeing the much larger versions of. Very cool. It's a kind of a new sighting for this area.
All right. There's more
Thanks.
to the right.
Ooh. This is helpful, getting a really good close-up. Of course, a lot of these you have to dissect them and look at the mesenteries, I think, to figure out different species. A lot of the identifying features are internal.
Thanks,
So we'll stick with zoanthid for now.
Pull wide. Lights are clear.
And you see, yeah, quite a few of these really large rockfish kind of looming in the background.
You see the ox one, Bobby?
Yeah, that's very, very close.
Survey 2, you see ox 1? You on it?
They're recording it now. What happens if we bring up the Ogs lights with the Ogs light camera?
Kind of blows it out, though.
Okay.
Gets in the way.
Hello.
All right,
He's trying to decide if we're bothering him enough for him to have to move.
Hello. All right. I'm gonna back away from the
You know, for most of my work, I don't usually pay much attention to the fish. I'm looking at the invertebrates that they're blocking. I guess it's kind of like trying to look at your rocks and all of this invertebrate fauna getting in the way.
Yeah.
Everyone has their thing, but I've definitely grown to appreciate them.
Let me get another strike here.
Oh, nice.
Sure.
Little partings, almost shaly partings, and that makes me
So you're at
think those are
waypoint 3.
either flow banding or perhaps that's a little interval of,
watch leads what they want to do again, but
sedimentary
at this point,
rock
we start
or
heading uphill.
Great.
perhaps tophaceous sediments that are part of the sequence.
Watch leads, nav.
Yeah, watch lead.
Yeah, we're looking for some direction here. We're at waypoint 3 now. Would you like to continue alongside or back uphill?
Yeah.
What's your preference?
Well, yeah. Let's kind of wrap around these outcrops. I could keep going a little bit here.
Roger. We'll keep following then.
Give me a 15-minute correction. Or, I need to go 190, right? Or am I mistaken?
Yeah, we don't need to strike it quite as much.
Right, Verona? Can we move it directly towards D2? Is that okay?
Sure.
Or is that a little too close to this? The fish?
Yes, it is.
If you see an octopus, let me know, bro.
So they want to keep going along.
I think you do like one.
Yeah,
I'd
just
go
a little bit.
I would just go slightly port of what you are.
180?
Yeah.
Alright, give me 15. I'll try to get my face back into something good here.
Actually, I'd go a little bit more. 170.
Alright. Great.
You want, what, 15?
170.
I'm getting some more tether watch. Did you see something here you want to zoom in on?
Actually, yeah. If you want to zoom in on the rock face,
10
that's
meters,
a
170.
little bit clear there to the left of the lasers. We might just have a little look at the rock.
Bridge, Charlie Nav.
We'll just look at this rock.
Can I get a ship move, please, of 10 meters bearing 170 at decimal 2?
You see this rock is
Good copy. Thank you, Bridge.
You get a sense of, again, it's again dark and fine-grained, but I see a little bit of horizontal streakiness, a little bit of banding. And on the assumption that we're looking at a volcanic rock that might reflect the flow banding from when it was coming erupting down the side of some slope.
cool. It's a big rock.
Nice big boulder. You can see it's more or less in place. You can kind of see how it the fractures and the joining in it, as they become more and more expressed, will break the rock up into what we later find as boulders like this as they start to come loose by whatever process around here, perhaps earthquakes on the Queen Charlotte Fault that have knocked that loose. Yeah, who's this?
That's squid parts.
Little squid parts.
Huh. could have cut the squid in half. Thanks, video.
It's good surface on that rock.
Alright, I'm gonna have to stay above this. I don't want to get the tether caught on that bad boy. Alright, Fernando.
Okay, coming up.
I'm gonna move it this way.
Alright.
More fish. There's two of them. There it is.
Go ahead, Bridge. Roger. Move complete.
Obviously, lots of good habitat and happy hiding places for the rockfish.
I'll go for squirrel next.
Little shrimp up in the water column.
What's the strike to the next waypoint? Bobby?
That's similar to my heading, right?
Yeah, next waypoint is right in line, 190 to Sirius.
Yeah, I think we can kind of keep on this heading as we head for the next waypoint. And we're going to crossing like what looks to be a little bit of a flat spot, although it's clearly pretty steep off to the left.
I think come left, see what's on the other side of that, and then we'll bring Sirius towards you. You ready for that, Fernando?
Yep.
15?
Yeah, 15 should be good.
15190?
Yeah, 190.
Bridge, are we nav? Ship move, please, of 15 meters, 15, bearing 190 degrees at decimal 2. That's a good copy. Thank you, Bridge.
Come in video?
Move in.
Wait for some tether. Yeah, hold that.
Okay, yep.
So a nice little forest of these hydrocorals, if I'm not mistaken, on this vertical face.
Yeah, what's that called on the zoom in? Oh, it was a tube worm that retracted its business. You know what I mean? You see that?
Those.
It's little tentacles. There's over left.
Huh?
It's over there somewhere.
What's that? I don't know what that bulgy thing is there.
Hold.
I guess
You
just
say hold?
looks like maybe one of those bigger tube corals.
I saw like a little tube go. Yeah, see it going, one of those. There's one of those. Just one of those things, yeah. I wonder if it's catching food from all the water rushing by it.
Yes, I would suggest that the featherworm is.
Featherworm? Is that what they're called?
I believe so. Colloquial.
Yeah, this is a great view of a tube worm showing its feeding appendages out there catching this marine snow drifting by. It's a really good look.
Right. We have a new commentator. Do you want to introduce yourself, Emily?
Hi, everyone. I'm Emily Ash. I'm a marine biologist, but I am working at NOAA Research Headquarters right now doing communications. So if you follow NOAA Research on any social media, I'm the one behind the scenes.
But you have a little bit of background in biology.
Yes, yeah. I've done a lot of research in mangrove ecosystems. Coastal zone management is what I have my master's in from UMiami. I've done some shark research. This has been on my bucket list for a very long time, so it's great to be able to be aboard Okeanos Explorer and be part of the deep sea exploration. I've been following along with these live streams since I was a very little girl, so living the childhood dream right now.
That's great, and Emily is also becoming very good at removing all the biology that encrusts the rock samples that we're bringing up.
Yes.
I know already she's going to be pretty busy.
Yeah, I am now the master rock scraper. Today's samples will be my biggest challenge.
Coming up over this rock window.
Copy. Delta's 15. Rotator 70 degrees. Coming up with you.
Co-pilot, your bearing to waypoint 2 is now more 205.
So is this one of the first expeditions you've been on?
This is my first expedition I have been on. Very exciting, hopefully.
Right now.
So
more to come.
What are your impressions? Is it kind of what you imagined, or?
Oh, it's everything I've wished for and more. There's so many cool things.
The long days.
Oh, they're my favorite.
And the difficult to sleep nights, or are you doing pretty well?
Honestly, I like it. Getting rocked back to sleep, to fall asleep, yeah. It's very nice. Let's see. Ooh, another rockfish.
Bobby, you want to introduce front row? No.
And Jen Ashoff has checked in with the chat with a very detailed description of the rock units here.
Why don't you take it, pilot?
Let me see. I saw the local name of the group.
Ooh, it looks like we have another soul.
I usually
soulfish.
trigger EX
Center screen.
Oh, yes.
for introductions.
You can come in, video. Go for eyeballs.
That's great.
Yeah, likely the Wales group. These would be Cambrian to Proterozoic, so that would be about 560 million years and older, or 500 million years and older.
Excellent.
Okay. I'll try to
Sequence of volcanic rocks and flows along with interbedded mudstones and shales that have been metamorphosed to fairly high grades. So we're getting some thinly foliated here, mostly nonetheless pretty massive rocks.
go for the shell item.
That's a nice view of the gastropod right there. Very nice.
He's got the parasite on. Going for the ride along. How you doing, Friday?
I'm doing good. Doing great.
Yeah.
I could,
Looks like you're lined up to waypoint 4.
Yep.
So we just gotta go up this. Okay. Next video.
Has anyone checked in with a call on those flatfish? Are they sole, or are they likely or are they halibut?
Hold on. Come out. I'll get you there. It's gotta be around here somewhere. There it is. No extra lights.
Yes, a lot of it.
that is 50%.
Take another 20. There we go.
Take more zoom video. I'll try to hold it here. Hold that. As you can see, I'm fighting a little bit of current. Alright, go ahead. Or zoom if you don't.
Adjust my focus.
Okay.
Wow.
Make somebody a good desktop.
Top part going. Hold that.
There's my focus.
There you go.
Play it on.
Clear the screen.
Sorry. All right, let it go.
Full wide.
Thanks. All right, he's lined up. 204. Just got to turn port to port.
Lights clear.
We could probably get 10 in, right?
Yep, I'm ready. When you are.
Oh, you just back on. That's a big one.
Roger, 10 coming in. Copy.
He's like tall, right?
Some more rockfish. Had a little eel pout swim by there.
Bridge ROV nav. Ship move, please. 10 meters, bearing 205. That's right, thank you.
And another one.
Let's go ahead and keep the lasers on.
Okay.
See a little bit of the foliation here and the rocks to the right.
Kind of got to wait for some tether rolling. You want to just take this fish while I wait for tether?
Sure. You should have tether.
I'm way out there, I think.
It's only 45 degrees.
Yeah, but look how small B2 is on your camera. Feels like I'm way out. Alright, Bobby? Yeah, I see it. Still there? I think we're way out. Oh, sorry.
We're in a little area where the marine snow is falling upwards. I don't know if that's we've caused that or if that's just a local current as the currents are running around these boulders and steep faces.
Full screen. Okay.
Kind of slowing down there. Maybe it was us.
Fill the screen.
Roger, on the screen.
Clear.
Okay. It kind of looks like brains, doesn't it? The encrusting sponge. Gonna snap it. Yeah, I'm seeing some. Okay, let it go.
It's a nice sponge.
Bridge.
Encrusting sponge.
Mm-hmm.
Alright, proceeding up this ridge here.
Move complete.
Copy, move complete.
Yeah. We're getting closer. And contact, come in. He's got a big foot, doesn't he? Foot to shell ratio.
That's great. A nice gastropod. You can see its mantle really nicely, its body under its shell. I'm not a gastropod expert, so if anybody knows what this is in the chat.
Yeah, I was going to say, did that all come out of that shell, or
Yeah.
is he on something? Wow.
Does that all retract in there, or does it stay out?
It can retract in there. They have this this thing called an operculum that's kind of like a trap door. So, but this is a great little view of this guy.
Thanks, Bear.
But
Roger.
yeah. Fun fact about gastropods and these sea snails, and snails in general, is their feeding structure is a radula, which is basically a tiny tongue-like feature that scrapes away material that they're feeding on, and it's covered in these tiny, tiny little teeth. And it's quite disturbing if you look at it under a microscope. You wouldn't expect such an intense tooth-covered tongue from your average snail.
Would not want to Arrowhead snail with 2 T-flags.
Are we still moving now? We're not, right? They called
Correct.
down.
They're stopped.
Copy.
I'm at 238.
So we're currently at a depth of about 340 meters. So we've come up, maybe a little bit more than 100 meters since the start of the dive up the side of this cliff when we're actually getting near the top. Climbing up one of the, at this point, we're about to ascend if we're not starting to, at the base of it, already climb up one of these last big ledgy cliffs that we see in the bathymetry.
Watch lead nav?
Watch lead?
Yeah, just to confirm before I hand off to the lunch watch, we're at 0, our last sample was 03 bio, and we're continuing uphill at this point to waypoint 5, correct?
That sounds good to me, yeah.
All right, thank you.
I'm going to go for the dark red guy, Ron, right below. See, where is it?
I thought you were
Looks
going
like we
for
have a
the
tiny little
squid
squid that
water
just went past at the top of the
Maybe I
left.
think.
I'm going for this dark red guy. Oh, it's a little baby, what's it call it? Alright. Nudibranch, what is it called?
Nudibranch?
Yeah, it is
Nudibranch, or
It's a little one.
a little sea cucumber, or a little
Yeah.
nudibranch. Very cute. It's
I have
very
a-
small compared to the other ones we've been seeing. It's
It's a little one. Sure.
Yeah, I don't know how to tell those apart.
Did you guys want to zoom in on
Yep,
anything else?
Christopher Moss says that is a nudibranch. It's one of the predatory ones. Very cool.
Tritonia.
Oh, you can see the, not tubers, what you call it, feather ones? Flying away. We're going to do a pilot change up here. I'll get back in a minute.
Sounds good. So we're having a little pilot change here, so we're just going to take in the view for a minute or two.
There's so much life all in one spot. So much diversity. Everything from your echinoderms with your sea stars to cnidarians with your coral and anemones, all the way down to mollusks with your sea snails. So much, all in one place.
A juvenile primnoa is being called out in the chat.
Looks like there's one of those larger predatory nudibranchs almost at center screen now on that rock base.
All right, this is Merlin back in the biology chair. What did I miss while I was out at lunch? Oh, that's you. There we go.
We saw a smaller version of the predatory nudibranchs that we've been seeing, the tritoni.
Oh, those big pink ones.
Mm-hmm. But this guy was a little guy.
Okay.
We did briefly pass over a white nudibranch I don't think we've seen yet.
Very cool.
But
All right,
just
Chris,
more of these
I got a couple of
rockfishes,
requests from you.
some eel pout.
Go for it. Let's put
Oh, I
by.
love those little guys.
Yeah.
shilling cam up there.
I see some nice juvenile primoas
Okay.
on this rock.
Too short.
Well, now you're in
Sure.
the geology chair.
Yep.
Well, need some help besides just rock.
Well,
you might be.
Instead of serious
Might be a little
cam,
slow in the rock commentary while Jamie's
just
away.
a little bit.
That's all right.
Get wire cam up and
You've been
monitor,
seeing lots of the same things, unless
whatever it is. 13.
unless he's requested a sample in this area. I think we'll just stick with rock.
Oh, over here? Yeah.
And let's get CPHD up instead of AF cam.
Some more of that kind of almost branching white
Nova,
Dema sponge that we've
LED
got a couple of nice close-ups
I've got
of.
CPHD to the right of it. You want it to the left?
The terrain in this area is incredible.
Yeah, I want the wide view.
Just everything covered in a
The
thick
wide view
layer
AF
of
Oh,
this,
the AF cam. Gotcha. Not the-
detritus that
Understood.
settled marine snow.
The digital AF, yeah.
Yeah,
I
what would
gotcha.
be causing these these mucus webs we're seeing on the rock faces?
Ooh, good question
think
that I
move that
don't
one
know the
left
answer
and put
to.
the
I guess
digital
there's quite a few mucusy animals that we've encountered on
is
the
true.
survey. We saw a hagfish a couple of dives ago.
Now
We
they're all up there.
know that some of the sea stars, there's one that's
got it, bud.
conspicuously referred to as the slime star.
That's true.
That seems
All
to
right.
like the slime things.
So
but
I've
it
got
could
you at
also be
4.5.
from
I love the new router setup.
farther away.
Do you want me to
or
keep you
higher
at
up in the
4.5
water
so you
column,
can
and
see
it's
where
just
you're
kinda
centered, or
settled,
do you
so
want me
some
to
of,
keep
like,
you
the
centered and I'll call out
larvaceans
the angle?
create those really
on the cam?
beautiful
We can
mucus
What's
nets
your
that are
preference?
filled with bits of
I'll do whatever you
detritus,
want.
and they
just follow me.
sink.
And if I get more than 30,
They're abandoned and
40,
remade
then
a couple of times a day.
tell me to quit.
So those will
popping
also
off. If I
sink and
down
kind of get
below
tangled up in
75
some of
or greater
these
than 75, tell me to
little
need to scoot.
tubes and
I'll
branches that
follow
are
you
coming
then with
off
the
of
cam.
the rocks.
How about that?
I think some of those long tubes of the tubularians we were seeing earlier, kind of big solitary
Coming towards center.
hydroids that make really long stalks.
Center screen. Below laser left.
Stand by. Let me get in there. Come wide.
See it?
Yep.
And a nice close-up. I always appreciate that, so you can just get a little bit of a feel for how much life is covering these rocks. Every time we zoom in, I see something new.
You don't want some fogs on that?
Yeah, lights at
ROV
about
watch lead.
40.
Copy.
Could we pan down to that kind of red fuzzy thing to the lower right?
Sure can. Do you want to see the star first while I've got it centered?
Sure, yeah.
Okay, stand by for white star. Go ahead, Roland. I've got it squared up and toes planted, more or less. Oop, toes drifting.
Ooh, just saw a fan from one of those tube worms snap shut. This looks like a really small Ceramaster, one of the cookie stars. And I still stand by the cookie star common name. I don't know about this ravioli star. I think everybody knows my stance on that by now, but I'll just
for
repeat it.
awareness, you got your lower stode, your upper stode, but you still have your shilling out.
Yeah, I think maybe a juvenile Ceramaster.
It's your call. I think they're
And
flattening
to the
out a
left
little
of
bit,
it,
so we
that,
can if you want.
encrusting white
Okay.
blob, I think, is a colonial tunicate. Kind of looks like a encrusting sponge, but I've been given those samples to look at under the dissecting scope, and they don't have any of the spicules that we would see. Oh, cool little eggs down there. Could we zoom in on whatever those are at the bottom? Maybe some little...
Oh, very cool.
Yeah, little eggs, perhaps.
Is that what we're looking at to the right? Or did I miss what you were asking?
Oh, very cool. Yeah, I don't fully know what those are.
Center screen right now?
Yeah.
What's this in the middle of them?
I think that's a scale worm. On the top, a polynoid scale worm, probably. But then on top of this encrusting sponge, there are these little globs, and I'm not totally sure what they are now.
Want to put the uppers out?
Yeah, it's always good to capture a nice full zoom of anything that we don't know, so that we can review the imagery later and try to give it an ID. That's great, thanks.
Okay, did you want to look at the pinkish red thing?
I do still want to look at the pinkish red thing.
Okay, when you come out, Roland, I'll just
Thinking
back
it might
off and
be
shift down.
maybe a nudibranch is my guess from way back here, but shouldn't guess. We have the ability to go and look.
We're going to reset. Resetting.
Uppers are out. Good. Okay. All right.
All deployed again.
Fogs are still up. We'll leave them that way, right? Okay.
Hydraulic secure.
Lots of these
Hydraulic
white
secure. Copy.
hydrocorals.
Go ahead, Ian.
Are really abundant in this area. Ooh. Very cool. Maybe, yeah, kind of seeing some of the features, those branched appendages coming off the back. That reminds me of some of the nudibranchs that we've sampled earlier in this survey. Definitely different from the Tritonia that we've been seeing. So maybe a couple different kinds of nudibranchs here. Could we get the lasers when we pan back just to get a quick size reference?
Okay, lasers are, I'm going to paint them.
Oh, also, can we zoom in on the other red stuff just to the lower right of this nudibranch?
Oh, and is that what you were talking about?
No, but now that I see it, I want to know what it is. Okay, I think some of those little solitary hydroids as well. Wow, look at the zoom.
Oops, stand by, lost
Very
my
cool. No, that's fine. Oh, another nudibranch down at the lower right. Yep. Wish there was something that could let people at home know what I'm pointing at. It would really help a lot. And lots of those kind of spiral, pyramid-shaped shelled snails, the Calliostoma.
Okay.
Very cool. Yeah, I think what we're looking at are a different kind of solitary hydroid. They don't have the really long, thin stalk. Ooh, wow. Oh, yeah, look like little flowers, and they have those little tentacles coming out from around the sides. And I think that also looks to me like a carnivorous sponge, that white thing, kind of tall, thin, white thing in the middle looks like an asbestopluma maybe. Little baby anemones. There's so much to look at when you zoom in. Maybe cereanthids. I see one of them has a tube over here.
Slow pulling out.
And of course, lots and lots of encrusting sponges. Some of these look like calcareous sponges, which are a group that I don't know as much about, but they're based with a skeleton of calcium carbonate, kind of like the corals, instead of silica like the rest of the sponges.
210, 205?
205.
Copy.
Very cool. Thanks for that zoom.
I may creep a little bit over this.
It's in the starboard rail cam. Did something gooey get stuck on the lens?
Videos.
I think we're
It
good
does
to
look like, yeah, some kind of seaweed or
continue along this feature.
Yeah,
oh, another
like
cool
slime.
egg. Maybe like a little egg trail attached to that little tube.
Full wide.
Wide copy. Shall we move along?
Lights are clear.
Copy. Pause off.
Yeah, would pilots like another move?
I think so.
205?
Yeah. Looks like we're a good spot.
How long? You want a 10-meter move?
20?
ROV watch lead.
Go ahead, watch lead.
Could I request a sample here?
Guys,
How are we situated?
We're just about to call in a
Just
shipment,
about to move?
so we will hold that.
Okay, yeah.
What do you see?
I've had a request for some cereanthids, and we just saw some in that zoom.
Okay.
We're hearing that.
I hear that name a lot, but you're going to have to help me.
Oh yeah,
Exactly
we'll
what
zoom
we're
back
going
in,
for.
and I'll point it out.
Okay. Is this going to be a slurp, most likely?
Yes, a suction for sure.
When you get anchored.
We'll just kind of try to scrape it off. Because
Maybe
it's on a
that
hard
millimeter
rock,
down
there's not
left
as
from
much
those
room for
red
it to
guys.
run.
Maybe a bunch
Sometimes
of
when I've tried sampling cereanthids in soft sediment, they just go, they disappear into the
John, I'm pretty sure we had
into
an
the mud,
imported
and you never see
sample
them
in
again.
jar 1.
That's
So
affirmative,
I think
yeah.
we'll
Go ahead and
just
start it.
bypass that. Yeah.
Jar 2, yep.
Yep.
We'll circle back there if we need to at some point.
Yep.
Got it spun up?
HPU. Hydraulics on.
Ooh, your first sample collection.
You want cameras?
No. Yeah, sure.
Oop.
Why not?
Oh, there's a
samples.
different
Okay.
fish than the ones we've been
Interesting.
seeing. That is interesting. Almost looks a little more cod-like.
Shore out easy, shore out. Shore out fast is fine.
That's cool.
4.6 is good.
So we have a bit of speculation about what the species might be here, but they're a very understudied group, and I think little is known about the cereanthids and the hydroids that we might be seeing. So
or you want me to follow you out to the tip?
getting a sample to study it in detail in the lab would be really useful to have from this area.
Okay, Roland, I know you were watching what we floated away from. Get me back there if you don't mind.
Just up left from that star you see center screen, straight up from lasers.
Yeah.
And then diagonally up to your right, past the pink. And now immediately right of lasers.
Okay, let me settle in, and Copilot, can you make sure I don't run the slurp into something? Go ahead and turn it off, please.
So there's a few things I'm interested in on this rock face.
What do you want me to turn off?
Oh. No, not. I'm looking at hydraulic motor, sorry. Make sure that suction pump's not running.
Probably get a couple of bonus brachiopods and brittle stars if I had to guess. They are everywhere. It'd be really nice to confirm the species. They look a little bit different from the larger dark brown ones that we saw.
So it's where those red things were, the
dives.
last things we
The red
looked
thing
at?
is what we're going to go for?
No, it's a different red thing in that zone.
Oh, all right.
So we'll just settle down and then get a
toe
nice
in and we'll
tight
zoom
zoom.
It may be the ones
Try to
actually
pick out
at the bottom edge of the screen. but the
organisms
ones
we're
I were
looking
describing
for.
was next to the...
Okay, you want to go partial?
Yep.
You can walk into it. Okay, science, you want to give a little guidance?
Okay, I'm scanning around trying to find what we were looking at before. Maybe up by the nudibranch, I think is where I saw it. Huh, maybe not.
So
Where did
go
they
down
go?
to the red's lower right.
Maybe scan to the right and to the left, looking for cereanthid anemones. So they are like a little anemone with a big tube. There we go,
Right
yeah.
there.
Oh, no, that might be well, might be, or is that a worm? Oh, yeah, there we go. You got it. So actually, I'm interested in all of this stuff, that little sponge and the hydroid that's attached, but the main target is that little tube with the tentacles coming out of it.
Okay, I'm going to go in. You want to go ahead at like 3 to 500 RPM?
Yeah, I can get it going now.
And someone needs to get the bucket cam, slurp jar cam up. Can
Wow,
you do
and it
that,
is
Nav?
small. Good catch.
We'll pulse it as needed so we don't damage him. I'll go in there, and you can stay partial, Roland, if you don't mind.
Yep, you want
Might get a
any
bonus
electron
heating math
HD2.
while we're at it. They're just everywhere in the water column here.
We're not quite that tight, please. And pilot, I'm starting to get close, so go ahead and crank it.
Okay, suction.
Take them all.
Go in,
Yeah, there's a lot
tighter,
of interest in
Roland,
this space. So we might have to kind of scrape it a
So
little bit.
that's
It's
suction at 700. Get you more.
just like you're vacuuming out your car.
That's 1,000.
Go in there. That pink thing, is that what I'm...
I think, well, I don't see it anymore. I think you probably got it. But if we could just do a little scraping around here, there's lots that we're interested in. Love to get a
See it in the
brachiopod.
jar.
Thank you. And that sponge just to the left, and some of these little hydroids would be a great collection.
Got some things in the jar
Are you
there.
okay? Should we back off on the suction, you think? Or is it okay, guys?
I think it's okay.
I'm not looking at the jar, I'm just...
They don't seem to be getting damaged.
Okay, good. All right, I'm trying to get that pink and the little miniature bivalve looking. There it goes. All right, what else? That little sponge to...
Yes, please.
next to the string. Okay, all right. Come on.
Oh, there's a scale worm in there too. In the jar.
There we go. I think we just got.
Yes. Oh, and what about that other sponge? Yeah. You think, is it within reach?
It's in the jar.
While we're here.
Yeah. Beautiful. Probably have 5 different species of sponges just in that little slurp.
Anything else?
Oh, see below the brittle star, that little polyp,
red.
reddish? Yeah.
Okay.
See if you can get that.
You okay, guys?
Yep. Samples look good in the jar.
Samples look good. Thank you, Nav. Let me see if I can
They're just
tickle
swirling around.
that little pink thing.
Oh, we might get a tubularia, a
He
bonus.
just went to hide. Let's see if he...
Yeah.
You can't... You can run, but you cannot hide.
Whoop.
I
Okay.
think I got him. Did I get
And
him?
then, what about
Arm
one of
that,
these
Brostar?
white hydrocorals?
We're gonna get the arm of that Brostar because we just hurt him, and we're just gonna. That way we hurt him on purpose instead of by accident. How about that? No, he doesn't want to let me take him.
Yeah, they're pretty. They're covered in spines. They get really stuck.
All right.
See that white hydrocoral, the branching coral? They're pretty brittle.
Down
But if we
low?
could snap off a bit, that would really help while we're here.
Okay, Roland, you with me?
Yep.
Maybe a little tighter. Okay, hold.
This has been on our collection
There's a
list for a
piece.
while. That's great.
That's enough?
Yep.
Okay.
Oh, a little,
crab.
yeah, a little decorator crab underneath. I think we'll leave that one.
All right.
Still watching for the last items to come through.
That's great. Very happy with that. How many different phyla are in that one sample jar?
Okay, last
we'll have to count them and let you know.
That'll be a fun jar to process.
Yes, yes. We're very grateful for your skills in the lab. It's been making things work go really smoothly.
I don't think there's any swimmers.
So of course, every different group of organisms has to be carefully separated out, assigned a tentative ID, photographed, and preserved.
What are these clear things we've been seeing swimming in the water column?
Oh,
a nice squid.
Yeah. Are they kind of like ball-shaped?
Cone, like stick-like. They've been kind of everywhere.
Stick-like. The
Almost
little,
like the very clear ones everywhere.
Yeah, arrowworms probably, or Ketognaths.
Interesting.
Yeah, yeah. I've seen them lots in a series of mesopelagic or midwater surveys that I've done off of the East Coast, but they're everywhere. Yeah, Ketognaths, also called arrowworms, and they have they're pretty much translucent, very small, and have two sets of really vicious-looking little teeth that kind of light up with a bit of iridescence. They're very cool.
Ooh, very interesting.
And they are everywhere. Might be attracted to the lights
the
here.
Lights, yeah. I cannot say I am a worm fan.
Say box
Oh, they're so small.
Sounds good.
That's okay. We have other worm fans here. There's something for everybody.
Watch lead now.
Watch lead here.
Can you confirm that sample was 04 bio?
Okay, sure.
Yes, 04B.
Good copy. And do we want to avoid going back over that suction jar in the future? Or you
Yeah.
want anything coming out?
Yeah,
Okay.
that'd be nice to have
We'll
it
go ahead and mark it as a
discreet.
potential swimmer. Thank you.
Thank you.
Yep.
And that's suction jar. Which, sorry, which jar? 2?
That was canister 2, yes.
Oh, sorry. Maybe I missed one while I was out at lunch.
Number 1, they attempted to suck a sample into, but it ended up just being dirt, and they ended up blowing it into the bio box. But we're going to, for now, mark that as contaminated, and we can circle back to it if we need to.
It looked like
Deal. Sounds good. Okay. Yeah, I'm all caught up.
Yeah. Oh, inside? Yeah. Little swimmer.
That was great. I think that is the biggest number of bio from a single.
I suppose since
sample
Jamie is
location.
here, I should probably log that. Let's try it.
I'm good on hydraulics. You got it clear, right?
Yep, hydraulics secure.
Okay. Swing arms are still looking sweet.
My sample pilots. My samples.
Good work together,
There's
guys.
a whole lot going in there.
Camera magic.
Right. Are you good with the jar? Do you want the still back up?
Maybe still can. Still can would be good. I'm going to do a scan on it. Yep, multi-beam. While I'm a little high here, let's see what's out there. Are we good? Watch lead on getting a move in.
Yeah, I'm good to continue.
All right.
15 meters at 205. Still good, pilots.
I mean, I could come over a little bit to square up, but I think if Todd's happy where he's at, that works for me.
205. All right.
Ooh, and that's a great squid right there.
Oh, wow.
Very well.
Maybe early on.
I love seeing it when they're...
Still camera.
Yes, I just did. Yeah.
Free HRV now.
Coming on.
Partial zoom.
Oh, yeah, let's follow this
Yes,
squid
we'd
for
like
a
to finish
second.
ship move if now's a good time.
Lights roll or no?
Yes.
Range
Beautiful.
15 meters, bearing
I love it when they
205
tuck up their
degrees at
their
decimal
tentacles
knots,
like
please.
that, their arms.
Is he getting away from me?
Using that fin to kind of fight the current.
Good copy. Thank you, Bridge.
So a lot of these are in the genus Gonatus, but that's just for context. I wouldn't hazard a guess at its identification. I don't often catch squid in the work that I do looking at the seafloor. This is very cool.
We've been seeing a good amount of them today.
We have, yeah. Oh, I love it.
It's really making some progress.
Yeah. Yeah, a little bit easier of a current today than in our dive site yesterday.
Are we clear?
Wow.
Oh, that was great.
Maybe bumping. Just
beautiful.
Coming up. I'm still chasing him.
Good.
There he is. Have we gotten any stills on him?
Yeah, they were snapping.
Good. I think I might have to let him go.
Wow, look at all this. Huh.
That's the direction I'm going.
Oh, there's a nice big Primnoa.
We'll let him float.
For the moment,
Coming back to say
unassaulted by sea stars, but a lot of its friends are.
Oh,
Whoa! Wow. Okay.
Here we are.
There we go. This is kind of
Look what he showed
a little bit of the abundance that I was hoping for, that the species distribution model was kind of pointing to. I'd love to get some good shots of this little localized coral thicket. Oh, yeah, another little tiny one, maybe one of the glass shrimp almost.
How are we on tether?
Wow.
Hard to see.
Lots of material for these Hippasteria. I'm not worried about the coral so much in this area. One of those really big nudibranchs on a huge Primnoa. Could we get the lasers on this?
Lasers are on.
There we go. Wow.
These are some great rocks, too. They seem a little bit more exposed than the ones we've been seeing.
There are rocks here, yes.
Yeah.
Yeah, I think the having the exposed rock seems to help for settlement of these larger
Do you have the
corals.
ability to pirouette around it?
I'm thinking I will, but I do have a lot of
That nudibranch? Yeah, there's two.
came up and over this.
Two really big ones. There's
So do
one
these
down there.
nudibranch that we see on these coral, are they feeding on them? Or are they just getting up above the rocks?
I believe they also feed on corals, but someone in the science chat,
Lights
please
up
correct me if I'm
40%.
wrong, but I've definitely seen them
I have full confidence in you, too.
on corals that have been look damaged and with no sea stars in sight, but
Great stills, too.
it's possible. I'm not seeing a lot of damage underneath of that one in particular.
Look at this
Maybe
rock.
they are
Look in
just taking advantage of the
Minipam.
extra height.
Look how
Oh, this is
fine this
beautiful.
feature
Yeah, this is really great.
Wow.
Very linear there on the ridge.
All right, I'm working hard on last to go sideways and keep it smooth.
So you can see why we're interested in these corals, especially the larger kind of arborescent or tree-shaped corals. They provide a really intricate habitat, a little bit of an extension into the water column for some of these other animals. Okay, this one has at least 3 really big sea stars on it.
As far as I can tell, you look good. I can't really see you.
Can you
rear
back
it off?
off as you go around?
So there have been studies done on coral bycatch and other samples looking at the number of associated species. If you start picking them and counting the kinds of organisms that are living in there, they can be hundreds on an individual. So often you'll see lots of different kinds of crustaceans, shrimp, and amphipods. We're seeing those mollusks again. Look at all that.
It's interesting to see such healthy ones right next to some other coral that aren't looking so great with a lot of exposed
A couple of them are having
stalks.
a real hard time, but still lots of living tissue.
Yeah.
Oh, wow.
Very good.
Okay, I'm going to let that float off screen, and we'll just come back around again.
Good reference, though.
We were doing a little bit of camera magic science just to get that shot.
Sure, sounds great.
There's a bit of current coming to my face now that we've come up on this flat area.
Yeah, I can see that.
I'm going to come back to the coral now. And if you
So the
want any
current is
particular
picking up.
view-
We're just going to
We can
reposition
see,
to try to
there were some-
get a good
bunch
look
of
at
sea
this
stars, the bigger ones.
it's
But
really
any of them
abundant
where
coral
we're
area.
seeing the underside of the star would be best.
Pilot, why don't
looks
you
like
turn on
definitely looks like a hat.
If you don't want me to, I'd like
I'm
to
thinking
turn
those
on
are
our
maybe demo
altimeter.
sponges. They're pretty floppy for any of the cone-shaped glass sponges that I
do
know.
have the BBL on as well. I can secure that if you'd like.
It looks like it's working now. Unless you see interference, we can maybe secure it.
All right, you're sliding back.
Okay, I'm going to push forward again.
Yep, so that's me looking at you, 7.5.
7.5, copy.
Good picture and multi-beam.
Good picture, multi-beam. Yeah.
Come down a little bit.
Look at the current. Look at what's changed.
I checked. I don't
Thanks
have
for
a
that.
sound
Thanks for
internet,
joining
so
us.
Let's see.
Your
Slack
visual.
tide was like 10 and 4 today, so we got a little.
Okay, so we're seeing some dead skeletons, pretty big ones of Primnoa, and then just a huge living colony on the top of this really steep rock feature.
Coming up to it.
And of course, lots of Hippasteria happily feeding.
Current might increase, is what I'm getting at, right?
It's we're in the middle of high tide coming in right now.
So we're fighting some pretty serious current conditions as we try to get a little bit closer to these beautiful Primnoa colonies.
Wow.
Look at that. Again, some funny comments in the science chat depending on if you're on Team Coral or Team Sea Star. But again, these are really slow processes. So Chris Ma was saying earlier that they followed the same individual sea stars a year later. They were the same 3 on the same coral. So it takes them a long time to kind of move their way up and completely denude a colony of the tissue. And these Tritonia nudibranchs, you've seen quite a few really big ones. Beautiful. So yeah, we're seeing a couple of the tips. There's a section there where there's some tissue missing, and these Calliostoma snails. You can see each individual polyp of the Primnoa kind of hanging in the water column. Sort of taking advantage of a little bit of space that's unobstructed by other fauna on the top of this rock and growing out into the current to gain access to food. I don't think access to food is hard to come by in this particular area. It just is full of marine snow. Oh, cool! Look at that barnacle maybe to the lower left. Something sticking out. We could zoom on that barnacle. It looks like it's doing something cool. Yes. So you see there's like a, wow, a little bit of a empty patch where this barnacle has found a place to settle. And of course lots of brittle stars. Those gastropods, the Calliostoma. Cool. Shrimp everywhere. Amphipods. And this barnacle also thinking about reaching out to feed.
Yes. Yeah, it keeps reaching out and then going back in.
Mm-hmm.
Is he just trying to see if he's got anything that time?
I'm not sure what that barnacle's up to. But it's very dynamic. Wow. So we see lots of the, dead skeletons kind of to the left and the lower part of the screen, and some really big living colonies along the ridge of this rock. Just beautiful. So again, this is commonly known as the Pacific red tree coral, or Primnoa pacifica. If we could just kind of scan this area to get an idea of how far it goes. Maybe to the left. But I know we're fighting the current the whole way. Seems to have picked up quite a bit since I came back from lunch. Wow.
Well, nice boulder pile. When I first got back, you could see that foliation or schistosity. Wow, that's hard to say.
Whew, yeah.
Right after lunch, I guess.
Another one of those words you read a lot but don't often say out loud. I have that problem.
Right. But a little bit different from the blockier outcrops we were seeing up on the cliff face, and makes me suspect we might be seeing more of that type of rock up on as we get up onto this little more flat area we're headed up to.
Okay, so we're kind of at the top of this feature, which is cool to note. That's where we're seeing the Primnoa. We're at about 330 meters depth. This is kind of right in the zone that they like to be. Yeah, it'd be cool to get a close-up of this sea star, although it looks like it might be taking a break. Just digesting.
In a few months, he'll go back to eating, it sounds like, by the timeline we got earlier.
Mm-hmm. And I do see more of the pedicel area on the underside of this sea star. And now I'm going to wonder for a long time what they might be used for. You can see this branch of a Primnoa skeleton just completely removed of all living tissue, starting to get encrusted on maybe by some sediment and some kind of tube-dwelling invertebrate.
And I forget what these are called.
Pedicelaria.
Pedicelaria.
Yeah, you can see a bit of the tube feet, and I wonder if that's a bit of the stomach. I know some of them kind of prolapse their stomachs out in order to feed. Beautiful. And there's some living coral tissue just down to the lower left. And little, all kinds of little amphipods. Little bits of detritus, I would think, kind of caught up in the branches. So they often have this kind of golden color, and then they kind of get encrusted with things and turn like a darker color over time. Beautiful. Those are some really nice, healthy-looking polyps to the left. I know they won't be that way for long, but, well, maybe another couple of years. Kind of in between geological time and normal biological time. There's a much slower rate in the deeper waters of the deep sea. Could we take a Niskin here?
I was just thinking that.
Oh, good. Yeah, especially in an area of such high Primnoa abundance. I think it's a good record to have. And I did enter that one.
Okay, so we're on 5.
Yes. Unless I missed one. Yeah, sorry, RV, I'm not sure if you heard me. Okay, great, thanks. Yes.
ID EX2306-D11-05W, Niskin Bottle 2.
Oh, interesting. Yeah, that another one of those Calliostoma snails and a whole tuft of these, I think they're tubularias, the kind of solitary, mostly solitary, although these look like they might be kind of connected and branched hydroids at the end of these long tubes. Very cool. Yeah. It's great. We did have a request of mollusk feeding. And lots of little amphipods, I think, crawling all over these little tubularias. I think I'm seeing some, maybe amphipod tubes as well. Saw some of those earlier. They're kind of mud-encrusted tubes that these amphipods make. Beautiful. Can we pan to the left while we're here? I thought I saw something cool at the base of one of these corals. Oh, wow, there's a rockfish just peeking out from in between those slabs. And up a little bit. Oh, no, over, yeah. So just to the left of that big sea star, at the base of that kind of healthy-looking coral at the back on the left, seeing something pinkish. Yeah, just a snap, just to get an idea. Can't tell if it's a fish or a part of a nudibranch. Aha, fish, thanks.
Oh yeah, good eye.
That's great. So maybe using the base of this coral for a little bit of a hiding place wedged in between the shelf of this rock. Thank you. Wow.
Well, it looks like each one of those corals has its own sea star.
Yes, at least one.
At least, yep.
Mm-hmm. So could we scan, like, is it possible to go to the left and around this feature just to get an idea of the extent of the coral distribution here? Okay. That's great. Just as much documentation as we can get of this area. It's fantastic. A real Primnoa thicket, even if it is under siege. Still lots of really beautiful, healthy corals. Wow. Oh, teeny tiny little squid floating, swimming through. Cute. Beautiful. Third time's a charm. I'm so glad that we, caught sight of some of these really large corals on our third dive in this area. Documented a few recruitment areas, probably with some juvenile corals, but nothing like these. That's great. Thank you. So we're getting an idea of a gonadid shrimp, squid. Some of those little squid that we've been seeing, definitely gonadids, but to be sure that it's gonadus, you need to be able to see some of the tentacle structure. Yeah, I am interested in this sponge as well. Still trying to figure out if it's on my list of species of interest, but I don't think so. Oh, possibly. Actually, do you think we could get a piece of this one? Thank you. Yeah. Should be, spongy. Not hard and brittle like some of the other ones. So I think we should be able to tear a piece. It also looks like it maybe drifted down here. Maybe not. Anything from kind of the lip, so I can get a good look at both sides, is also always helpful. And kind of the transition between the two. I think it should. then what will we do with it? I think, can we zoom in on the base just to see? I wonder if it fell down here. It looks like it's kind of toppled a bit. Oh, I see. No, okay. Maybe just a piece then. If we can kind of rip out a piece like what we do, yeah.
Is he anchored to that, or is that just his foot?
Looks like, yeah, the basal attachment has grown pretty solidly on that rock, and that rock doesn't look like it's moved recently, so I think it's just kind of slowing in the current. It's a really large individual. I'm used to seeing them more upright. This one does look like it's kind of bowled over to me. There are a few different demosponges that have this kind of shape. One of them is on my target list, so I think it would be helpful for a collection. Oh, the cup coral. That's okay to mix those two if you want. Cup coral should be there pretty solidly. Maybe a little less. Yeah, and we can just snip. Yeah. Yeah, that's fine. That would be great. Should. Of course, it's always a risk when we don't know what kind of sponge it is, how it will behave. There we go. Great.
We're going to go port outer.
Beautiful. Thanks for that collection. It's excellent. Yeah, that's what I have. Mm-hmm. Okay, so it looks like we're continuing to follow this kind of almost, what would you call it, a ridge-like feature? Not quite. It's another sea urchin. Oh, and some of these little, very linear silvery fish. Unfortunately, all of our fish biologists are off for Labor Day, so we don't have a lot of great fish IDs. A lot of invertebrate biologists represented in the science chat and here in the biology seat, so we'll just have to kind of capture the observations for analysis later. More Primnoa skeletons and a little bit of living tissue.
And you see those little poorly kind of rounded off ledges in the outcrop, which I would guess at this point would be the schistosity and the layering of this rock, and similar to those boulders that we saw here, a little more in place.
Oh wow, looks like we're coming up to more Primnoa. So we see a lot of the kind of dead skeletons or partly dead, definitely being stripped of their tissue by sea stars, the Hipasteria, some of these Calliostoma, snails. Could we actually zoom in on the snails again? Yeah, just another observation of the snails kind of eating. Beautiful. Wow. Yeah, the current's just kind of ripping through here.
But forever, well, I see plenty of particles. I was going to say, not quite as turbid as it's been, but not true.
So we've
Right. We were barely seeing the lights, yeah.
Mm-hmm. The reading is down in the 1.3s. Yesterday I think we had a 1.6. Yeah. I think I'm seeing maybe some of those little amphipod tubes. Definitely a lot of these Calliostoma gastropods. The kind of pyramid-shaped shell in profile. Still lots of living tissue of the Primnoa. Little amphipods everywhere. Of course, these larger, probably pandalid shrimp. Beautiful, and the kind of golden-colored skeleton of this coral, where it's been recently had the tissue removed.
Oof.
Look at all this life. Really big hip area on the other side. That's great. Thanks for that.
Now, as we zoom out, I'm sort of looking around at the boulders. This wouldn't be a bad place to get a sample of this more apparently more foliated rock if there's something nearby, and I've got a couple rock boxes still waiting for me. Don't see anything immediately. Maybe one of these, yeah, and one of those in the foreground or coming into the middle. Yeah, either the one that's kind of straight below where the lasers would be, or perhaps the one that's over in the left corner, lower left corner. Might be a good size and something that's easily grabbable. Yeah, the little thing. Or even that one with the dark face is another good one too. Yeah, let's see if we can get that one. Have a look. Looks like it's not too big. Yeah, and then we can have a yeah, no, that'll be all right. And it's got plenty of stuff on it that that's a little bit.
Wow, there's lots of stuff on it, actually. Something over there, I don't know what it is.
And is it possible to just zoom out just a little bit and see what this one to the left of him looks like? I think that looks like the same thing. Yeah. Yeah, that looks like a good one. Sure. Sure, yeah. Right. He's moving pretty quick for a sea star. Or is he a brittle star?
A brittle star, I'd say.
Oh, that's why he's moving so fast.
Yep. And I think, woo!
Makes us escape.
Yeah, that's a good plan. Smart little brittle star. Looks like there are a couple more still attached, so we'll see what we get. Oh wow, a little bit of everything. Even the bottom. Whoa.
Wow.
Just ripping through here. Okay.
Yeah. I missed where we put that. Was that the port or starboard side?
Starboard.
Starboard. Sample ID EX2306-D11-07G. Starboard forward rock box.
Can we pan a little bit to the right just to get a quick view of some of these large primnoas? Beautiful. See some more even farther in the background. But we are getting really pushed around by the currents, picked up quite a bit in this area. Wow, that one is huge. Can you just get the lasers on that one to the right? Can try. Yeah, it's okay if it's a little too hairy. Wow. Oh, there's K2, I think.
Oh, there they are.
Wow.
Looks good.
Beautiful. Thank you for that. Aw, look at that.
Half a meter wide.
Wow.
Maybe a little more.
Yeah, they're pretty giant. Okay, thank you. Whew. With pretty much every primnoa you see a very happy looking Hippasteria. Lots of those little silvery fish along the bottom here too. Wow, even more primnoas coming out from the shadows. Yeah, I think it's a rockfish. It's kind of the back. Or lying down, maybe, I don't know. Oh yeah, these big Pacific Dover sole, I believe. This one's a little bit grumpy looking to me. Looks like there's a bit of damage to this one, actually. Look at the top there. Maybe he got bitten by something. Wow, ouch. Wow. love if we're here for zooms after this. I would take a look at that really nice, healthy-looking Primnoa beside us. Thank you. Wow. Can really see the torsion in this one, the mouth kind of twisted sideways, and the whole rest of the body with the eyes kind of in the same place. Wow. Whoa. Look at the range of those eyeballs. I didn't realize how much the whole thing could move.
After scanning around, he's decided it's time to get out of here.
Just kind of done with this close-up. Wow, look at that. You can see the spacing between the teeth is kind of big. Whatever took a chunk out of that one. Ouch.
Ow.
Yeah. Looks like it's healing, though. Yeah, another one of those interesting kind of small stalked sponges as well. I think there are a couple of species that I've seen in my guide that have that morphology, a couple of different demo sponges.
Getting a little bit more sedimentation here. You see the sediment piled up on just the one half of that rock, but not enough to discourage the coral.
Mm-hmm. Yeah, I think
He's
once it
stuck to the more vertical face that's not getting sedimented.
Mm-hmm. Yeah, and I'd say once they get to a certain size, the sediment at the base probably bothers them less. But yeah, this one up in the foreground, kind of pristine. It looks like it hasn't been predated on by any of these sea stars. It looks perfectly intact. It's just beautiful. Wow. Yeah, really healthy looking, kind of fully extended polyps of this Pacific red tree coral, Primnoa pacifica. I think I can see little eyes of shrimp kind of peeking out. Yeah, this one's beautiful. Very healthy looking colony.
Do you think that being anchored out here on this kind of solitary rock surrounded by sediment is maybe a little bit of protection from the predatory sea stars?
I'd wonder. I could imagine one of those sea stars, starting to crawl over the soft sediment and getting swept away because there's not as much to hold on to.
Right.
Right. So if they're used to kind of associating with these bigger rocks, then maybe they would need that to kind of grip. So maybe it's on a little bit of an island in the midst of all of this. It could be kind of acting as a little moat.
A moat, sediment moat.
Yeah, to defend against these coral grazers. Little amphipod, I think. Crustacean attached to the end of this branch. Wow. And then all the, of course, all of the polyps and the coral behind this one. There's just so many here. Really big individuals providing habitat and a little bit of extra complexity and structure. Some sort of slow zones, kind of creating little eddies for smaller fish and invertebrates to hide and live. Just collecting some still imagery of this really beautiful coral specimen. Yeah. Gives you just a bit of a feel for the kind of abundance that we have in this area. This is fantastic. This is what I was hoping to see when I saw the coral prediction model just light up in this area. So I'm very happy to see all of these Primnoa.
So what kind of things go into these models for predicting species distribution? Is it potential substrates, or as well as current?
Yeah,
whatnot?
Definitely like a combo. So the first thing that they will bring in is all of those bycatch records. So I think the one that I looked at used about 20 years of coral bycatch data from the trawl surveys. So every time a trawl touched the seafloor and came up with a bit of coral, it was documented, weighed, I think, or counted, identified to the best of their ability. So some of them are lumped, and then those records were ingested into this model along with, I think I saw, depth for sure is a really, big predictor in coral distribution. Temperature, and I think I saw something like current. I'm not sure about substrate, if they brought in any kind of mapping like that. Actually, I could check. I was just looking at this ocean color, so different measurements of the seawater, maximum tidal current. Whoa, look at this one. Yeah, those seem like the main factors that were sort of ingested by that model to give us this prediction. And look at this huge coral. If we could get the lasers on it for a nice measurement, especially at the base. Wow. So the base alone is over 10 centimeters wide, closer to 20
Wow.
Part of it. Just huge. And we can really clearly see which way the current is going, and it does look to me like these are kind of growing in a way that orients them to catch the current and catch all the little particles that are coming out. There's a nice Acanthascus sponge at the bottom that looks pretty pristine as well. But look at this one. Just when I think I see the biggest, healthiest looking one, then we come and see this. Just beautiful. You can see all of the particles kind of going through and around these branches, and you can imagine each of those tiny little polyps, reaching out into the water column and grabbing bits of detritus and all of the organic matter that it feeds on. There are lots of, some really pristine looking ones up here. Just beautiful. Oh, I can see a fishtail in there. There's some kind of fish that's hiding out.
Yeah, we are coming up to another little, although it looks pretty flat on the high-pack bathymetry, another little ledge, a little slightly steeper area, not as steep as what we saw earlier, but will be a little bit of a rise.
There's a big sea pen straight ahead, kind of at the top. And more Primnoa, I think just the bases of them kind of coming into view. Yeah, I'd love a close-up, please. I think we've seen a little bit of this one or a similar species. It's Helipteris or Balticina, I think is the newer name for that genus. So a few different species. This could be Helipteris californica. There's a pretty distinct one, a different species that I don't think this is, but then there are one or two unnamed species from this area. But samples of those exist already in the museum collections to my knowledge, so we'll just get some nice imagery, especially close-ups of the polyps are always very helpful. You can see little spines at the bases, should be a pair of them, I think, that can help us kind of give an image-based ID. And of course it's getting whipped around in the current. That's great. I think that's yeah, I have a good look at the spines. This is enough for us to assign it a morphotype, maybe a tentative ID. Thank you. Oh, sure. Nice view of that brachiopod and some brittle star arms. Lots of little tubes from worms, probably. The stalks of those tubularias. Little sponge.
The little, what do you call a brachiopod, little fingers?
Oh yeah, there's definitely some tissue on the top here. You mean these coming out?
No, more in the internally.
Well, I haven't looked at
tentacles.
I almost call them cilia.
Yeah.
But that's just me. I'd call it cilia. That's what it looks like to me. But yeah, little tiny hair-like structures. Probably helps with filtering out food that it's interested in, is my guess. A loaf of 4. There we go.
Loaf of 4. I always thought of that as more of a singular thing, but that's from way back in my college years, taking paleontology.
Oh, okay.
Right.
It's like, what biology? Ah, paleontology. Wow, another giant Primnoa. A couple of damaged or predated ones, but still just huge, huge specimens. That big one right in the middle there. Wow.
So we've reached the top of this, so 100, 150 meter or so high rocky face we came up, and we're now up on the summit at a depth of about 320, 325 meters, which is where we'll be, I think, for the rest of the dive.
I mean, I'm okay with this. I'm okay with being in and around here for the rest of the dive. Look at all these beautiful Primnoas.
There's still a couple ridges up here, but they're only a few meters high. And
ROV, can we zoom in a little bit? There might be a bit of fishing line. Just a straight line, kind of, well, actually, could you turn the lasers on and I can direct? So it's like the line in between the corals, just above the lasers. Yeah, still a little bit above, and it's going, yeah, left to right there. Just passed over them, I'm not sure. Could just be another kind of rocky feature, but it looked a little too straight to me. If you pan to the left. Oh, maybe not. No, the closer I look, the less I see it. Could just be, yeah, some debris kind of following a crack in this rock slab. Thanks for the zoom. Wow. Look at these just giant Primnoas. Look at that one. I think I see more coming out of the shadows, so another area of high abundance. These Primnoa pacifica. There's a little ctenophore floating into view. Well, pretty big one actually. Yeah, I just love to follow this along as much as we can and document its extent. I'm sure scientists will be interested in kind of calculating the size and the overall health of the corals in this area. That's what I'd be looking at if I was annotating this video anyway. Watch lead here. Yeah, sure.
Absolutely,
yes.
yeah.
Are you? Yeah. Yeah, I mean, if this ridge is where the corals are, then I'd love to follow it. Lars, you missed a skate earlier. Oh, good. Oh, good. Good, good. I'm thinking a long nose skate. You know, I haven't had time to look it up. If somebody sends me the still of that, then I could probably narrow it down. Wow, look at all of this. So again, we're seeing lots of these Hippasteria sea stars, but even more abundance or number of these Primnoa corals, which is fantastic. We'll follow along this ridge and kind of try to document the extent. We've had some really beautiful close-up imagery. Now I just kind of like to see if we can kind of count them, get a feel for how many are here. Every time I think we've come to the end, then I start to see more coming out of the shadows. This is fantastic. I'm happy to follow this ridge.
From the perspective view, it looks like we might have a little bit higher to go to the top. There might be one more little ledge to climb if the if what I'm seeing is right.
And do you think those are corals pinging, those blobs, or are they rocks? Could they be corals?
I think they could well be.
Wow. Wow, so yeah, these large corals just kind of dominating that area. They're just everywhere, and it looks like there might be more coming up ahead. We're starting to get little signatures that in the sonar that normally I would have thought were rocks, but they could be these corals.
Oh yeah, so here we are. Now we've moved. I'm not used to the scale of being this zoomed in to the 3D model, but yeah, now we are. It does show us right on top of that ridge, right on the tip top.
Oh, another flatfish. Less sure about the idea on that one. So we'll leave it to the fish biologists. Whoa. So I'm also seeing lots of these hydrocorals. We were able to get a sample of one earlier, which is very helpful. ROV, can we zoom in on that white just below the lasers to the left a little bit? Kind of a blob. I just wasn't quite sure what I was looking at. Yeah, just a snap. Oh, okay. Yeah, a hydrocoral from a different angle. Thank you. Whoa. Well, there's our first Liponema, I think, the pom-pom anemone that we saw lots of a few dives ago. We've seen a little bit. Oh, just a lighter colored one. That's cool. Oh, it's really beautiful. Kind of a pale lavender color Liponema anemone. Oh, and some little tiny cup corals too. Very cool. Every time you zoom in, you see something different. And look at these really big primnoas.
There's another one, maybe a meter across.
Wow. Definitely some of the larger individuals that I've seen.
Please pardon the interruption. Your conference contains less than 3 participants at this time and will be...
Good to know. It's like, did I do that? What button did I push?
I never know, yeah.
Okay, so seeing those little hydrocorals at the base and some of the shrimp. I think some hydroids, those little kind of really small branching tree-like structures. I see yeah, now that we're closer up, little shrimp on the coral, on the branches. Some small anemones growing on some exposed tissue. Beautiful. So that's that part of the older coral skeleton at the base. Doesn't have any polyps on it. It's kind of like the trunk of a tree, but it's still covered in looks like living tissue. This one's a really beautiful, kind of pristine, healthy looking primnoa coral. I think we'll pause here to get a bit of imagery of this one. It's just spectacular. You can see the marine snow floating in against the dark background.
Kind of a wintry scene.
It is, yeah. It has been reminding me of actual snow quite a bit, especially just at so thick in this part. And the corals with their branches extended, almost making, practically like a wall for the water to slow down when it encounters the branches and the little polyps in between, and slows down the water and the debris that it's carrying so the coral can feed.
Yeah, from the 3D perspective, it looks like it'll be fairly flat for the next 50 meters or so, and then there's another one of these little ridges similar to one we went over, and one slightly smaller one on the way, maybe 20 or 30 meters out.
RV, can we get the lasers on some of these? Just would love to be collecting approximate height measurements of some of these corals as we pass by. So a reminder for anyone watching who's not familiar, those laser dots are exactly 10 centimeters apart. So you can use them to make estimates of the size of the things that we're seeing in the video. A few more of those, probably Strongylocentris genus of urchin. Sea urchins at the bottom. And of course, evidence that a Hippasteria has probably been here, judging by the condition of that coral in the middle. But And also there's some really beautiful, healthy colonies.
Do we know
Just
what the
everywhere.
range of these corals are?
I do. Where's my guide? Oh, another one of those sea cucumbers. The one's related to the giant California sea cucumber.
That his droppings that are right behind him? That's an interesting bunch of pellets.
Like it. Yeah, I'd say so. So that's Aposticopus is the genus, which is kind of a fun one to say. And leucothele, or leucothele, I think is the species on that one. Of course, the giant California sea cucumber being another species in the same genus.
That one rounded boulder we're just passing on the left, strikes me as maybe being a dropstone from an iceberg, since we see nothing else like it out here.
So I'm still just trying to get a feel for how far south we see this species. Certainly we get lots of it in BC waters, and I know the type specimen, so the original coral that was used to describe the species in the first place, was from off of Japan. So both sides of the Pacific, but as far as how north and how far south it goes, off the top of my head, I can't remember. Definitely, just a bit south of us in BC. We see them quite a bit on a couple of our seamount study sites.
Yeah, I think west is roughly where we'll get to that next little ridge, where there might be a little bit of downhill before then, though.
Hmm. Do you think we could pause for a Niskin here? Just while we're close to such an abundant Primnoa area. It's always good.
I have us on Niskin bottle 3.
Same. Perfect, thank you.
EX2306-D11-08W. There's going to be a little lump out there. Maybe about 2 meters high, but you're going to have to go down a little bit before you get there. Down maybe 3 meters, looks like.
Look at the view behind us.
Oh, wow. Things sure change when you get out in the mud.
Yeah. If we can see behind us, there's a big boulder covered in those from NOAA. A couple of different sea stars associated with the soft sediment. Another flatfish, possibly a Dover sole. Pacific Dover sole. Nope, there's another one.
Something looks a little different about that one.
Mm-hmm.
Maybe it's just the angle. It looked a little longer and slender.
Yeah, the ID guide that I have is kind of limited to halibut and 2 different kinds of sole. So it might not apply. Oh yeah, definitely. Could be just a younger version, those 2 side by side. They definitely look a little bit different. Oh wow. This one looks like the coral got so big it might have toppled that rock by catching the current. Yeah.
Now, would he be able to get back upright, or is his stalk too stiff?
It looks like they're trying. See, the tips there are growing.
Are starting to grow up.
Starting to grow vertical,
suggest he's otherwise stuck like this.
Yeah. Yeah, I wouldn't expect the part that's already grown to be able to change, but then the new growth can go up. Of course, as soon as it gets a little bit heavy, I think it would start to sag, but actually, yeah, the ends of that coral look pretty healthy. It's another thorny head rockfish, I think. A couple of them here. Yeah, this little flatfish side by side.
And different colors.
Yeah. So I think one, could be kind of a juvenile version of the other, or they could be different species. The, coloration on them do vary quite a bit. You have here, and show you a few different pictures.
And these are all Dover soles. Interesting.
Dover sole, yeah. So this is still my guess.
All right, yeah.
Unless somebody tells me differently. Yeah, it's kind of interesting to compare these two different, kind of morphologies. So, and another fish.
And
this one
there's...
we still don't know. Not great that I can recall. Maybe catch another one. This is cool, though, to compare the two morphologies. Kind of yellowish. The range on those eyeballs is very cool. Very cool to see.
So I kind of get the feeling I can see how they move their eyeballs around. Their eyeballs are completely surrounded by soft tissue.
Mm-hmm.
And so it's really almost more like pulling the skin around and the eyeballs come with it.
Yeah.
I always wondered how the skull deformed itself to move an eyeball around.
Allow that, yeah. Well, there must be, some kind of a basal region for the, ligaments to attach to.
Right, yeah.
Huh.
And what's that really bright-looking
The golf
spine?
ball?
Is that the golf ball? Yeah.
I think that's actually a colonial ascidian. So there's some very spongy-looking tunicates that kind of grow encrusting in encrusting forms like that, and that's what it reminds me of. Could be a sponge, but I'm just from the samples that I've looked at in the past, I've had those come across my microscope, and I've always been surprised. Look at it. Oh, this is not a sponge at all. So yeah, unfortunately, all I have to go on is kind of different color patterns, so the slightly different overall shape to the fish, but then kind of looking at those white dots, but color patterns on flatfish are apparently very variable.
Now this one's next to a tennis ball.
Yeah. Well, I think that one is a sponge.
Okay.
For sure. Or, well, it could be the back of a brachiopod, I guess. And lots of this really delicately branching pink hydrocorals. Just everywhere, wow.
Mm-hmm.
And some big primnoas up to the right. Yeah, if we see that fish again, it would be nice to get another zoom on it. I'm not sure that we captured it. Oh, great, yeah. Oh, there's two.
Couple, yeah.
Ooh. So I think I also maybe avoided this because I don't know exactly what they are. They're not in any ID guide that I have, and I don't think I've heard any fish biologists tuning in today.
Oh, Michael says maybe an Alaska pollock.
That's a pollock?
Not that one.
Okay, good. I was about to say, it's like I've seen pollock in the Atlantic. They're a little bit bigger. We thought maybe a herring earlier, but
it does look very trout-like.
Mm-hmm. And trout, okay. Oh, and another one of those stalked yellow sponges. It's always cool to see. Look at all these really big primnoas. Yeah, just a really small version of that one. Huh.
I don't suppose they could be juvenile salmon.
You're asking you're asking somebody that mostly studies sponges and corals, so I'd have a really hard time. I get a feel for the ones that are in my study sites, and unfortunately beyond that I'm not very helpful.
I know my only experience is perhaps trying to catch them.
Mm-hmm. Wow. Swimming to kind of stay in the same place, there's still quite a bit of current here. Beautiful, thanks for that. Someone will be able to ID that fish from this excellent video. Fortunately, not I. Wow. Again, another contender for a size record on this dive. That Primno is giant. Huh. Yeah, I think this is another one of those sea pens. Halipteris, maybe? Which I keep on forgetting is the old name. Balticina is the new name. It's been recently reassigned. Cool, so there's a kind of a larger swollen end on that branch called the peduncle of the coral that's anchored into the sea floor, kind of inflated with water to keep it there. Oh, we've had an ID on those little fish, Eulachon. I'm glad somebody's out there that can help us. So you can see each individual polyp of this sea pen, kind of like a different version of the polyps we were seeing on those really big corals. They're both octocorals, so if you were to count those little tentacles coming out, you'd see 8 of them. But the Penitulacea are in their own group, and they all kind of have the same form of one long central, pen-like stem with the polyps coming off of it. Often see them associated with soft sediment with modifications to bury the end or the peduncle in the softer sediment. Wow, look at all these really huge primnoas. A bit of kelp degrading and caught up in the branches of that one. A really big pom-pom anemone, that Liponema we've seen earlier. A couple of the Tritonias, and lots of these little lacy hydrocorals in the pink and white, probably separate species, most likely Stylaster. Wow, just beautiful. Seeing a little bit more predation on the bottom of some of these. One of those nudibranchs. Some kind of thinner, sparser looking branches in this forward one, which is really interesting next to the kind of typical version. I see the foot or an arm of one of those big Hippasteria sea stars.
Oh yeah, on the other side.
Yeah.
Yep.
I knew they were in here somewhere.
He was hiding, but
yeah. So it's really interesting. I often wondered if kind of the thinner, sparser branches wasn't a different, maybe there's a variant of this coral that I haven't seen myself, but it looks like it's all attached, so probably the same one, just with the tentacles a little bit droopier maybe. Very interesting. It's maybe just different stages of growth or feeding. We have a collection request. Is this an okay spot if we were looking for a Primnoa piece? Just a snip of any Primnoa you like, as long as it's got some nice tissue at the end. Yep. These red kind of tree-shaped corals. Well, I suppose kind of orangey pink, actually. I think it could mix with starboard outboard if you wanted it to, but it looks like starboard inboard is also free. But I think it's just a rock, so it shouldn't get mixed up. So again, we took a small piece of Primnoa on an earlier dive. This one will be really helpful to have for comparison. There's something very interesting going on with the population genetics of this large habitat-forming coral, so it's of extreme interest to scientists to be able to take a look and compare as many samples as possible from this area to others from around the Pacific. And of course, any slight morphological differences that we might see that kind of correspond with those are always helpful when trying to decide, what the extent of the species is. With that information, we can have a better idea of what kinds of vulnerabilities they may have. It's always helpful to have as much information on their distribution as possible. Mm-hmm. Sure. Is that the little arm cam? Yes. Oh. Oh, sweet. I see it. Oh, now I see the screen that it's coming out on. I love it. And just, a small snip is all we need for our purposes. Thank you. Yeah, I want a copy of that arm cam footage from here. This is very cool. See a pretty big flatfish flying through in the background. Yes. Yeah, maybe. Yeah, that's great. Thank you. And they're pretty tough, so anything that's not, snipped might kind of hang on. if that helps to know. Beautiful. I don't mind a bonus Niskin in this area. Whew, beautiful.
You told me just in time. So we just did a Niskin? Okay, so I have the Niskin as sample ID EX2306 D1109W, Niskin bottle 4 at 325 meters. Starboard. And the Primnoa EX sample ID EX2306 D11-10B in the inboard bio box, starboard inboard bio box.
Beautiful, thanks for that. Well, filled once. Perfect. Done our Niskin duties today. It's really easy to get distracted and forget. I could find one.
And you still have all those canisters left.
Yeah, I think I have an idea too. Wow. So this is the abundance of Primnoa that I was really hoping to see. It's very exciting to see that this model has done a really good job at predicting where we might find an abundance of these corals.
And in this high-resolution bathymetry, you can even make out these little mounds that are no more than a meter or so high,
right,
that they're on, as opposed to the flatter, more sedimented areas in between where they're still present, but a little more few and far between.
Huh. Yeah, I haven't been keeping a close eye on this because I've been so focused on the corals, but that is a really interesting observation to note that they're, less on the kind of sides of these cliffs where I was kind of thinking they might be, and more on these little bumps. Oh, that's okay. Yes.
Right. Mm-hmm. Yeah.
Okay, yep. Sounds good. So we might have a little bit of space from the bottom as we kind of traverse this dip. Then we're approaching a wall-like feature, so we should see more shortly. So you can, really easily see which way the current is flowing, carrying all of this marine snow. Still seeing little Ketignaths and shrimp swimming by. More of those little silvery fish. I think they are Eulashan. So it's interesting. I sent a little bit of info about them. They're anadromous fish, which means that they move between fresh and salt water. They're a kind of smelt, which is one that I'm a bit more familiar with in some of the small rivers in PEI. found from northern California all the way up to here in southwest or southern Alaska. Wow, another huge Primnoa and an almost equally huge Hippasteria. Huh. So we have a bit of time waiting for a ship move. We could take a good close look at what we see growing here. Wow. So you can see even some, dead and damaged polyps, those kind of whiter ones that are full of debris. Look like were maybe damaged by this sea star. And then some really beautiful kind of pinkish, living and healthy looking polyps around it. And a very sharp transition between where the sea star's arms can reach and where the coral is happy. See that really beautiful kind of golden skeleton of the Primnoa underneath. Some little shrimp. Every now and then I can pick out the eyes, and then I see there's a shrimp attached to it. Otherwise they're very well blended in. And a really good view of the Pedicelaria. Again, Chris Ma tuned in earlier to tell us a little bit about these structures, and they're kind of unknown in function. Not all sea stars seem to have them, from what I can tell, and they can kind of open and close. So they're a little bit mysterious, maybe for protection from bigger sea stars that like to try to predate on them, but they're on both sides, on the top and the bottom. And it's just speculation. Scientists apparently still don't know. I'm seeing lots of brittle star arms and lots and lots of hydrocorals at the base of this really large Primnoa. Some more Primnoa over here, and hydrocoral and a little sea urchin. Maybe a bit of a dead skeleton, some, I think a piece of kelp maybe. We've been seeing some kind of stray pieces blowing around. It's my guess, unless, I was like, unless it's a rock. No, I'm pretty sure it's kelp.
It's maybe up against a rock.
Mm-hmm. A really big Liponema anemone off to the right. Could you maybe zoom in on that dead coral skeleton with the tissue kind of around it? See at the base of those two rocks? Yeah, just a branch, and I can't tell if that's coral tissue or something else kind of growing around it. Yeah. Okay. Oh yeah, it does still have some tissue intact, so it looks like that snapped right off from somewhere, a bigger colony. Huh. Very cool. All right, thanks for that. Beautiful. Like the some of these I think were called the candy stripe shrimp. Not sure if this is the species that someone in the science chat was referring to earlier, but definitely see the really stark, distinct striping on the legs and a little bit on the carapace. And probably a Henricea sea star beside it. We've had a... Oh, look at that snail. I almost didn't see it.
Why?
Wow, it can really move.
That is fast.
Yeah, that was fast for a snail.
I thought at first I thought it was something
rolling.
Yeah, whipping around in the current. But there's another one to the right.
Oh yeah! Wow. Yeah,
they're very well disguised.
No kidding. This covering of debris made them nearly impossible to pick out, from the rocks beside it. It wasn't until we saw that one really move.
Or are those rocks?
Or are they? Now I don't know. It could be. We've seen urchins covered in all kinds of debris. We've seen decorator crabs, I believe, with a little bit of tissue on the top. And a little tiny, cute little fish right at the bottom, maybe something like a snail fish. So we have Fusatriton oregonensis, I think is a tentative for the ID for that gastropod we were looking at just now. So we're just doing a quick shift switch with our ROV team. We'll just pause to enjoy the scenery at 325 meters depth here in the in this part of Cordova Bay, just about to approach another one of these kind of lower, less steep wall-like features, I think. Excited to check them out for possible Primnoa colonies. Yes, love to take a look at this little fish. Oh, they're so cute. And a little tiny anemone beside it. Wow. Kind of looks like a little snailfish. Yeah, I don't think it's an eel pout. I don't think it's a grenadier or a rat tail. It looks like maybe a little snailfish to me, but no, it's fine. Ah. Oh. I was like, the fish that's right in front of us. I don't know. I mean, it's got a long whippy tail. I don't know. We haven't seen any rat tails on this dive yet, so it'd be very interesting. Cute. We did see a skate, though. Wow. Huh.
Wow.
Just beautiful. Let's see. Where? Is that a skate maybe? A chimera maybe? Let's go take a look then. We need... Yes, please. Ooh, a really big brittle star. But yeah, I'm also interested and invested in this fish ID now. Where did it go? Just saw a little glimpse of it. Oh, I think it might be... Oh, let's take a look. I do. Could be a chimera. Well, I just see it in the other camera. Sorry, I'm cheating.
Right.
Is that starboard? Okay. I'm going to pull up all of my fish guides.
Is that him?
Yeah. Ooh. Oh, it's so hard to tell without the face, but it looks roughly like the chimera. Let's see. Whoa, beautiful. Yeah, that's definitely my... Oh, these are beautiful images. I'm just pulling it up now, but yeah, it looks like a chimera fish to me. It's not even in my guide. They also call these ratfish sometimes, or I see a ghost shark. It's a cartilaginous fish.
He's going to see what's going on with this soul.
Cute. Thanks, we break for corals on this vessel. Wow, beautiful. Yeah, we definitely haven't seen one of these. I'm pretty sure Caitlin mentioned this this fish exactly as being very excited about. So I hope she's watching. Wow, okay. So yeah, this species definitely isn't in any of the guides that I have, but one of the chimeras. So I guess, oh, just beautiful. Closest living relatives are sharks and rays, though their last common ancestor with them lived nearly 400 million years ago. Species are largely confined to deep water. Very cool. Just beautiful, really unique. And look at the like iridescence in the lateral line. And the markings along the face under the eyes is so something I haven't really seen in any other kind of fish. Oh, they're so cute.
Yes.
Wow, look at this. Oh my gosh. Oh, wow. Really bright. Okay. All right. Maybe taking a bit of an aggressive stance. Really wanted us to
tired of us
there.
coming after him.
That's great. Sure. Wow. Thanks for chasing that one down. That was a very cool catch. I never would have seen that coming out of the starboard cam, so that was great. And doubly made Caitlin's day, I think. Another note about the chimera, they're very abundant in Puget Sound along with six-gill sharks. very cool. Sorry, Chris, we got a request for zooming on that brittle star, and I just, we saw it ages ago. Definitely got distracted with that charismatic fish. But if we see any more, I think he was talking about those bright red brittle stars. We'll definitely take a closer look next time. And more of these huge Primnoa colonies. Could we get the lasers on this one just to get an idea of the size? There they are. Thank you. Wow. Yeah. Sure, I'm happy with that. I could look at these all day.
This seems like one of the taller ones we've seen.
Mm-hmm. Yeah, definitely. I think each of these probably grow a little bit differently depending on the specific currents that they're exposed to. And for some reason, this one decided that, you know, up was a little bit more advantageous than growing out. Beautiful. Just the really deep orange color and the, you know, kind of black water column shot in the background. These are really stunning. See lots of these little arrowworms kind of flickering around in the foreground and the background in the water column. And just a really impressive abundance of all of this marine snow. Wow, these are so beautiful. Oh, okay. Oh, there he is. Yeah. Wow. And according to Elena Jorgensen in our science chat, there probably is a male chimera. Not sure how you tell the difference, but that's a good observation to have. Wow, we're seeing a couple of different things now. One of a really big boot sponge or rossellid sponge. A couple of those large sea pens, the Balticina. Liponema pom-pom anemones. And a few really large colonies of Primnoa pacifica. Some of them have fallen over. It may be a different species of rockfish than we've been seeing. We've seen some white dots in the pattern on these ones. Huh. Wow. Beautiful. yeah, every kind of ridge, area of exposed rock, we keep on seeing more of these really massive Primnoa corals. Look at this.
Wow. This is the best forest yet.
Yeah, this is amazing. Could we get the lasers on just to get some broad measurements? No, that's fine. Thank you. Where are they now? Oh, thanks. There we go. Yeah. Just to kind of get an idea, maybe around the base of some of these. I'm just, sure, thanks. Just thinking ahead for the person that might want to annotate this video. They'll probably have to count each individual coral, and they might even be asked to do their best to measure them, so try to help them out. Wow. Look at all these. Yeah, I'd love to scan this area just to get an idea of the extent. Thank you. This is fantastic. So yeah, I think we'd call this like, there aren't any hard and fast rules, I don't think, but maybe coral thicket. Just a really dense population of these.
I like
Primnoa.
coral forest.
A coral forest, sure. Yeah, I mean, if you're the size of a sea star, it is a forest. And even some of these fish we've seen kind of associating with these corals. Every now and then we've kind of scared one out. Sure. Wow. Really big rockfish coming into view. So you can see why, yeah, an area like this is of great interest. You see lots of smaller rockfish kind of scattered, especially close to the bases of some of these corals. But you can see how they would just completely change the dynamics here, give smaller animals a place to hide or rest, and maybe even brood. Wow. You know, and we are seeing if some of those Hippasteria sea stars predating on the corals, but in general, a lot of these are pristine. A couple of different sponge species in between, and lots of these, almost, they look miniature compared to the Primnoas, but these little hydrocorals, also in pretty high abundance here. Wow. Yeah, I would say so. Yeah. So yeah. Sure, yeah. So this is yeah, the area that the coral distribution model that we were going off of to kind of use for maybe a site selection when we were picking out this place, you know, took in all of that different data of catch records from bycatch, from fishery surveys, as well as a bunch of different pieces of environmental data. And it did highlight this area kind of as the high point, you know, as one of the darker red spots out of the whole Gulf of Alaska. So we had a pretty good idea, or high hopes, of seeing corals here. And if not, it was really useful to have to kind of ground truth this model. But we're definitely seeing what I was hoping for, which is, you know, kind of these really dense thickets or forests of Primnoa pacifica, which was one of the species selected out or the primnoid corals, these larger structure-forming corals that the model was looking for and looking to kind of describe the distribution. So this is kind of giving us a lot more information about the small-scale distribution and where we might find these corals in other places where we can't get to with something like an ROV. We can use it to fine-tune the model and help us predict places that might be good habitat. So I'd say this is a really dense coral population that the model predicted would be here, and now we're seeing it. Now we're seeing imagery of it. It's been a very, very useful observation, and just incredible to document such a dense amount of these really large, long-lived corals. We just keep on seeing more.
So seeing a little bit of lithologic difference here. Just we initially came up a more massive outcrop where we saw the big forest which is I believe to the left here on the view, and then over here to the right we see a ridge that looks a little more. It's got traces of bedding, or perhaps those are partings from the schistosity or the foliation in the rocks, and not so many corals on it.
What is schistosity?
Schistosity. Schistosity, yes.
Schistosity.
That's refers to the texture of a schist. It has that, we call it a foliation, but that general flat parting that's due to the minerals that make it up. And it's interesting, the little hydroid corals seem to stick pretty close to the bigger primnoas. ROV watch lead. Yeah, I believe we're kind of to the top of the last ridge, and we're going to be going down if we keep heading west at 270 for a while. We might want to make a right turn and head kind of northwest up this ridge, and eventually there we will kind of top out as well. But that might be the best way to go. Yeah, we might want to continue down this a little bit, and at 270 for a little bit till we get to the end of what I think is the outcrop, and then we can maybe turn and head north from there. Although, yeah, like I said, we would be heading down, so that might not be the best way to go. Yeah, I can see almost immediately, yeah, it's getting dark. So, right. So yeah, this might be a good time to make a turn to the right. Yeah. No, that's good. sea cucumber?
Yeah, that looks like a great sea cucumber.
Is that what they're called, or is that just a really good one?
This is just a really good one.
Okay. Sure.
Yeah, that'd be great.
Nice trail of droppings.
Yep.
Doesn't look like he's made a whole lot of progress.
No.
Kind of just hangs out right here, and he's found a nice place. He must be
Beautiful
finding
texture.
plenty to eat.
I would imagine there's a lot of little organisms in this sediment or debris on the top of the rocks that he could eat. Earlier you were asking, Jamie, whether the difference between a nudibranch and a sea cucumber. A nudibranch is a mollusk, so basically you can imagine that as like a sea snail without a shell, while the sea cucumbers are echinoderms, and they have, usually when you hear echinoderms you think of the radial symmetry, the pentaradial symmetry, and sea cucumbers, you don't see that on the outside as much, but inside their breathing structures.
Oh, interesting.
Where
Okay.
you see that symmetry. Yeah, but basically these guys just cruise along the bottom and filter through this detritus and sediment, and yeah, little vacuum cleaners on the bottom of the sea.
So if I think of it as kind of a one-armed sea star, inaccurate as that might be in detail, it'll help me remember.
What's this here?
And that looks like a
crab?
decorator
crab, yeah. With
a decorator
anemone
crab who's
on
decorated
top.
himself with an anemone. An anemone.
Very interesting. Yeah.
That's interesting. I wonder how well that actually works for the crab.
Yeah.
Or is he just hiding underneath of it?
That could
that's
be.
true too.
Yeah, maybe he's just hiding underneath there. It's funny, you pan out and you see, oh boy, not much going on, but you zoom in and think we're passing through to the other side, one of those, that coral forest we saw earlier.
Yes.
That sea star has eaten the middle out of that one, but
Oh, there goes
Ctenophore?
a jelly.
Something just out of view to the left. Don't know.
I thought I saw a big line in the Sirius view. I wasn't sure if that was our tether or a fishing line. We have an identification of a sedipid ctenophore that went through the view at one point. Mm-hmm. Oh, wow, yeah. Yeah, this really deep kind of trench feature.
Very nice, yeah.
We're seeing, kind of steep rock and coral on either side of us with lots of sediment kind of filling it in between.
Is that a shrimp at the bottom?
Where?
Or no, that's hydroids or something stunned. I couldn't figure out what that was with the arms sticking up way into the water, but it's a hydrocoral with something next to it.
Yeah, I think there's the base of an older dead coral stalk and some other little things growing at the base of it. Thanks, Michael, for joining us in the science chat. We've really benefited from your observations. Oh, wow, yeah. Oh, very cool. It'd be cool to go over top of them. Yeah, in the sonar view, we're seeing 3 really big stripes kind of returning. Mm-hmm.
Right.
Okay.
Yeah, I think those are, it's reflecting the, it's a compositional layering in the rocks where you're getting a little more resistant ledge and then interfingered softer parts and that are the low spots. I'd be interested in getting a piece of some of this stuff, especially where you can see that foliation or that bedding, those alternating ledges, but there doesn't seem to be much around here loose, like nothing.
If it was, it might be kind of in between, and it's just full of all this sediment and detritus.
I think that last rock sample we got was probably this stuff, but being that it was out on its own, you're always never quite sure, but that's also why you grab what you can get when you have it. So.
So we're seeing lots of these Hippasteria sea stars now. Got a cute little fish kind of at the bottom, maybe another one of those sculpins. So a lot more damage from predation or grazing by this sea star, and more of those kind of pyramid-shaped gastropods, Calliostoma, that we've been seeing. ROV, could we zoom in on that stripy fish to the left of the lasers? Yeah, is that what that is? Ooh.
It's a stripy...
Wow, with the little, mask, little banding around the eyes. Very cool. That's something a bit different from what we've been seeing.
Yeah. Is that another one to the right?
That one looks more like a rockfish to me.
Okay, yeah.
But this one, there's a fish I know that we've seen in some of our other imagery called a war bonnet, or a decorated war bonnet, Chirolophus. Wondering if it's maybe something like that. Yeah, it could be. I think I might see a little bit of a tuft on the top. It's kind of where it gets its name.
Oh.
Yeah, those little kind of embellished appendages that are protruding above the eyes.
Well, those probably sensory?
This is my
or just decoration?
Very vague guess. Yeah, I would guess that they have some kind of a sensory application, but if they're really spiky, I suppose it could be for defense or to blend in a little bit. It does resemble some of the hydrocorals we've been seeing, I guess. This is beautiful. So yeah, nice view of the patterning. The only other times I've seen it, I'm looking at the pictures in my guide, is also in association with Primnoa. So it's kind of an interesting one. And a couple of different polyps in behind it, some maybe a different kind of stoloniferous coral. It's hard to see, it's barely reaching out from the layer of sediment and detritus that's on these rocks. Wow. Again, you get a sense of how impressive the zoom is on this camera. Something that small. Could we get the lasers just for a general scale? That's great, thank you. Wow, quite a bit of damage to some of these Primnoa colonies. And I think by now we know who the culprits are. But lots of really big, teeny tiny squid. Very cute.
Another rockfish.
Oh yeah, a couple of them around here.
Couple.
So it's possible the snails we've been calling Calliostoma. We've just had another suggestion for an ID of Acoya platinum. So a different genus, but in the Calliostomid group. Calliostomid. There we go. So if we have questions about this snail, please let me know in the science chat if that's something that we should potentially target, because we haven't so far. Of course, there's always a chance we got one on the rock, but feel free to type in if that's something that you'd like us to collect. Otherwise, I think we'll continue scanning. Yeah, we've seen those. Those are those little sculpins. Thanks. So thanks to Chong Cheng for that identification at Jams Tech. Not sure what that acronym stands for. I'm just barely getting a handle on the Canadian government acronyms, so you'll have to forgive me. Wow, some really dramatic, almost like boulders with these geological features at the ends.
This is almost something we might be able to find a sample at the bottom of if we can get to the bottom of it.
Oh, there we go. Japan Agency for Marine Earth Science and Technology. Thank you for the help with the acronym. Oh, could we get a zoom on this white, the white color morph? It's not something we often see together. A nice big flatfish swimming by. But yeah, if we could zoom on this white individual. Thank you.
So that's the same thing, only in white.
Yeah, a really stark difference. There's an area, one of our study sites, that has almost exclusively the white variant, and it's always very interesting to see.
Oh, look at that ctenophore going by.
Wow.
That was neat.
Or a siphonophore, maybe?
Siphonophore,
totally
I'm sorry,
catch it.
yes.
I am also just starting to learn the difference. I brushed up on my pelagic IDs in anticipation of our midwater dive, and so I'm a little more on it than I was at the beginning of this survey, but not by much. So it's always helpful to document when we see something a little bit different, so we know that there's a variety between a white color morph of Primnoa pacifica and the really dark pinks, oranges, and reds. So it's very cool to see them, right side by side for comparison. We can see a similar polyp structure, the kind of highly armored polyps of these Primnoas. You can see almost individual sclerites, or those hard calcium carbonate skeletal structures embedded in the tissue of these corals. And there's another one of those amphipod tubes, those little tiny crustaceans that are making those little mud tubes.
Oh, they're making that, not just sitting there?
Yeah, I believe
interesting.
So. Yeah, this isn't something that I've ever seen before, but I think Sean Rooney piped in about it early on in our expedition, that it was a thing, something that he sees quite often. And now we've seen it in situ a couple of times. Beautiful. Yeah, look at all of the colors. Very interesting. just to see it almost could be the same individual. You know, we can't really, there's so many different attachment points down at the bottom, and they are known to grow, you know, newer colonies will kind of grow on the old stalks of dead ones, so there's a lot of overlap. And this one's relatively intact. It hasn't been predated on to quite the degree of its neighbors, so this is a really beautiful, you know, healthy-looking colony. Yeah, this is fantastic. Just getting the gradient of the different colors with the kind of dark open water in the background. All of the marine snow floating by, and like a storm of these ketognaths, these long little arrow worms that we see kind of flicking around in the water column. Just stunning. Wow.
There are tons of them. If we're seeing this many, I'm wondering how many there are right up in front of the lights, which
Yes.
is where they really like.
Yeah, every now and then we pan up and to catch a glimpse of what's up there, and they're just like moths. Oh, another one of those. Yeah, I think it's something at least very similar to the decorated war bonnet is the common name for this fish. The species I have
interesting. Yeah.
Yeah, so they really seem to like, you know, the Primnoa, very closely associated here. Wow.
Neat.
Yeah, so I think Chirolophus decoratus is the species name that I have for one of these fish that we've seen, you know, not too far away in BC. So we could be looking at a different species, but it's definitely something like Chirolophus decoratus. just looking up, checking out its hair. You look great.
Oh, remarkable.
Oh, these are beautiful. So again, a really great example of how these corals create habitat for other kinds of organisms. Here is a really big fish that's just nestled right into the branches.
And of course, Chris Ma is only noticing the out-of-focus starfish, sea star.
Right. I tried pretty hard not to call them starfish, but it's good to know that that's like a perfectly acceptable common name. I think most people refer to them as starfish, knowing full well that they're not fish.
Right.
That's beautiful. Even this kind of pan back is really stunning. There's the arms of another sea star at the bottom. Not sure if it's the same one or maybe something like a solaster. Beautiful. Another little squid swimming on by. Possibly a gonadid. I think a little jelly, that one might be the Solicinus. A little dinner plate jelly. Wow, look at the marine snow coming down here. How's the current in this area? Yeah. Mm-hmm. Okay. Mm-hmm. Yeah. Yeah, it seems to be really coming down here, the marine snow. So we've got a little bit of a dip here ahead of where we're going. So get a little bit of a view of the water column instead. Of course, we've been seeing just thousands of these little Ketignaths flickering through the water column.
Yeah. Yeah, there's a little indent, but...
Oh, lots ahead of us. There we go.
We can see it on the sonar.
Yeah, the sonar is really lighting up, so not too far ahead of us. We're seeing another big rocky area. Lots of little fish down there, I think. Could be myctophids or something else.
Yeah, I like that.
So we've been given an ID of Euphisa anthomedusa for one of those jellies we saw earlier. Thanks for that, Dougal. Lots of Ketignaths and amphipods. And also possibly a Euplocamus, another species of comb jelly or ctenophore. Okay, so coming up to another one of these large rock features we were seeing lighting up in the sonar, and again, it's just covered in some really large Primnoa colonies. Smaller rockfish hanging around. Some kelp caught up in the base of one of these corals. And a lot of those kind of yellow splotches on the rocks could be the bases of older dead Primnoas. Could possibly be some encrusting sponges, but they look like they could be Primnoa holdfasts from large ones. And of course, some of these smaller rockfish may be juveniles of some of the larger species we've been seeing. Wow, look at all these. ROV watch lead. How do you feel about trying to suction sample some of these little snails? The snails that are kind of eating the coral tissue. There are some on the ground as well, if those are easier. I'm seeing lots.
They're
the little
to
pyramid-shaped. Yeah, anywhere that you see them. I think we will try a collection if we have some suction jars open. Yeah, that's kind of what I was thinking. And since there's a little bit of confusion about what genus and species they might be, I thought we'd pause to collect maybe the ones on the floor, and then we could suck up a couple of little extras. What do you think? Yeah, I think so. Even though I'd love to help the coral out a little bit, that's not my job.
Plus, you'll wind up with a bunch of bonus brittle stars.
That's right. I'm trying to make the echinoderm scientists as happy as we can. We've really benefited from their expertise and their identifications on this survey. Well, good. We have a second vote for collecting some of these. Yeah, kind of take your pick, and then maybe we'll collect a little bit of extra encrusting stuff around it. Beautiful. That's the one I would pick too. Nice. Confirmed. That is the target. So again, these are possibly in the genus Calliostoma, but we've had another tentative ID of Akoya platinum. So it'd be very helpful to get a sample to confirm. Can be a bit more precise when we talk about the distribution of these snails and, you know, what they do, what they eat, and the kinds of environments they're found in. Can help us gain a better understanding of the dynamics of these ecosystems. So these snails will make their way to the Smithsonian, where they can be accessed from people pretty much anywhere in the scientific community who are interested in borrowing them. And they can be further studied based on their morphology. So the physical characteristics we can see under the microscope. And also for genetics, so we'll be taking a tissue sample and running them through the barcoding process, at least if there's not some kind of specialized application, and we can compare them to other specimens of the same species and different ones to see how closely they are related to each other.
There he went.
There we go. We'll keep an eye on the jar and wait for... there it is.
And there it is.
Beautiful. Yeah, a couple would be great.
There.
Oh, there it is. Yes. Beautiful. Maybe one more for good measure.
There's one.
Yeah, I see them. Nope, doesn't matter to me. Take your pick.
Got him.
I think so. No, I think that's perfect. Thank you. That's great. The Smithsonian collections also, thank you. I've, getting some excitement from the science chat about that one. Oh. So apparently, at one point, Akoya platinum was in the genus Calliostoma, so they're all obviously very closely related or easy to confuse.
All right. So I have sample ID EX2306 underscore D11 underscore 11B in canister 3.
Beautiful collection. Thank you. Okay, so I think we'll continue to explore this area and maybe with the lasers on when we can, just to kind of... Are they? Wow. Okay, I was like, I cannot find them in this. I do see the line, yes,
Right.
yeah, I do too.
Is that some kelp down low stuck in a
Yes, I think
coral?
so.
Yeah.
Beautiful. There we go, thanks. Could we do a snap on that kind of cucumbery, wormy looking thing under the coral? Right to the right of lasers. Okay, another one of those sea cucumbers. I think it was Apostichopus, something like that.
Wow, yeah. That was the last one that was really long for a sea cucumber, I thought.
Mm-hmm.
Are these any loose rocks over here to the left? Kind of, well, now coming into the middle. Wondering if maybe we could grab that guy that's coming right into the middle of the lasers. I think the lasers. There's also one that's now coming in towards center as well on that little sediment chute.
Mm-hmm. They're not making it easy for you. Aw, cute. Oh, there's 3 of them.
Yeah, that one looks, I don't know, yeah, as good as the other one, maybe a little blockier, and I think it's, well, already I can tell it's bigger than I thought it was as we get closer with the lasers, but I think it'll be all right.
Might have a Primnoa stalk attached to it, which would be very cool.
Oh, now it's getting bigger and bigger.
It is. Well, What do you think?
It's a maybe. Is something that's not too big. I think it's all right.
What do you think, RV team? Will this fit?
Yeah.
Okay.
Yeah. Then that looks like it'd be a good one. I think we need to go into port side.
Mm-hmm.
Yeah.
Yep.
Port side rock.
Wow, there's also a huge nudibranch on a dead coral skeleton behind. Kind of really blends in with these corals.
So these samples will help us do a little bit of geology out here in the middle of the bay, where in the last mapping that I'm aware of that was done, the contacts are left hanging as soon as they got to water. But the geology obviously continued down into the water, and this will help us kind of bracket. There's kind of two choices of rocks. The rocks to the west of us, the islands to the west of us, and the rocks that are on them are a little bit younger, Silurian and Ordovician. The rocks to the east are a little bit older, and Cambrian to Proterozoic in age. And with some isotopic studies, oh yeah, look, we're going to get that. Stock, maybe?
Excellent. Could potentially do an aging study.
Right.
If we have the base of that coral.
And we can maybe tell the age of the rock. And doing so will tell us a little bit about the structures that separate the rock type that in between the islands on the east and the islands on the west of us.
Beautiful.
Very nice. Sample ID 2023. I'm sorry. EX2306 underscore D11 underscore 12G.
Port rock box.
In the port side rock box.
That's correct, yeah.
Yeah.
Doesn't bother me. Yeah. I understand.
Crunch, yeah.
Yeah, no, that's totally fine. You never know, they might come up on the instrument tray later. I've had stranger things happen. It's another one of those Henricius, the white little sea star to the left with a red dot in the middle. So they're nice and easy to pick out. And I think we might have a different kind of sponge also behind where that rock was. It looks like it could be a Faria glass sponge of some kind.
Oh yeah, let's zoom in, see what we can see.
EX2306 D11-12G. Oh, great. Yeah, definitely a Faria of some kind. It's pretty sedimented. I think it's struggling a little bit here. But, yeah, one of the thin kind of branching tubes and lobes Makes you think it's a Faria. There are several different species in this region. That's great, thank you. You see them often associated with the sponge reefs, although they don't have quite the same skeletal structure. They don't create quite the same enduring large reef-like structures that the Afrocallistidae do. But very cool to see. I think that's the first one in this area that we've noted. Wow, just keep on going. Look at these. Oh, could we get a zoom on these little starfish in the foreground here? That red one, starfish or a big brittle star? Kind of can't tell. We had a request for a zoom earlier, and I think we got distracted chasing that chimera, so this is a great opportunity. Yeah, I have no regrets.
Oh, have we seen one of these
cool.
before?
I don't think so, no. I wonder if Chris has any thoughts. Yeah, just the interlocking those kind of big scales that make up the dermal layer of this. Okay, we have an ID of Stegophora nodosa. So I think if we know what it is, that's not a sample request, but some really nice in situ imagery is always really helpful. You can get all the way out to the tips. Apparently they feel like cookies, which is helpful information to have.
Interesting.
I bet they don't taste like them. So it looks like we've got a little bit of damage to the arm that's at the pointing at 12 o'clock. In situ video is very useful. Chris has always wondered what they do, and now we have a bit of an idea associated with this soft sediment and staying very still. Wow, look at the texture on this. I call it the central disc. You know, that round part in the middle. It's just beautiful. And yeah, lots of little, polygons, pentagons, hexagons kind of making up the geometry that you see. That's wonderful, thanks.
And before we zoom too far out, I'm kind of interested in the rock that's up and to the left. So we need to back out a little bit.
Science, this is on your science channel. That snail on the left edge of the screen, if you look back at the video later, that actually jumped and started swimming in the water column and then fell back down there.
Oh my.
Oh, okay. Apparently we missed a swimming snail. That is cool.
Dark looking thing. Yeah, just a quick zoom.
It's a creature walking on it.
Looks like a spider.
It does look like a sea spider, a pycnogonid. Different species from the Colossendius, which I practiced pronouncing a little bit after trying to before. But a different species, but definitely a sea spider. And maybe...
Alrighty. That is good. Right.
It almost looked like a piece of a bamboo coral skeleton next to it as well. But I haven't seen any on this dive yet, so it would be hard to say. Wow, look at these. There's even more. Just giant. Whoa, okay, the view from Sirius. Everybody should definitely go over to stream number 2 to check it out. You can just see
That is really good camouflage.
A little bit of camouflage and protection, yes. Yeah, I'd love a snap zoom on this. A sponge. Bit of a different one. Let's see. Ooh, a different kind of Feria. This one looks like Feria oca to me. It's kind of the largest species in this group. So it's kind of easier to pick out. And maybe another sea spider inside or something inside. Very cool. Oh, wow. And a really buried, it's just been covered in this sediment of Primnoa that's been knocked over. Wow, lots of Liponema, these big pom-pom anemones just everywhere all of a sudden. And a couple of those Balticina sea pens in the foreground. And just, I love seeing that imagery of from Sirius looking down on D2 as it's exploring this really beautiful, seascape. Well, I think all of the coral scientists that have been following along and will probably be reviewing this video in the next 24 hours will not be disappointed with this dive. We've seen, fields of these Primnoas. Yeah, I think based on all of those environmental factors I talked about and, kind of extrapolating those from the different kinds of catches that we've recorded across the Gulf of Alaska, predicted this area as a hotspot, and we didn't have any actual evidence until now that it's proved to be pretty accurate. This is definitely an area with an incredibly high abundance of Primnoa pacifica and lots of other things. But
it pays to explore. Huh?
It sure does, yeah. It's very helpful that I know we had to change our plans, and this was kind of a backup, but I couldn't be happier with this plan B. We ended up seeing a hugely abundant coral area that I'm sure scientists will want to keep an eye on in some way going forward. Okay. Just getting a little bit mesmerized by this ctenophore that's in front of us. But this is why, we do the kind of exploration that we do. You can sit with your algorithm all you want and use these predictions, but to actually be able to see and, count and measure all of these individual corals is something else. If you were ever looking for a study site to study Primnoas, this incredibly important habitat-forming coral, you couldn't ask for a better one than the one that we just discovered today.
Yeah, this is just kind of taking it to where you need to take it. Models are great, insofar as what you can do with them, but in the end, you really need to be able to validate them and see how useful they are, so you can move forward from there.
Yeah. Yeah, so you could, the area that the model covered was the entire Gulf of Alaska and reaching out into this into these little channels a bit. So you could never visualize, you could only ever do a very small fraction of the amount of ground that covered. You could never visit at all the amount of people and time and energy and equipment that go into a dive like this is pretty astounding.
Sure.
So you have to kind of find ways of narrowing it down, and the models do that.
Well, that's why the models are so valuable, for sure. They do. But that's why, at the same time, it's important to know that they're working right.
Yeah, and this one was, pretty much spot on. So I think some of the authors that did all of that work in 2017 will be very interested to see this kind of come to life. And also interesting where we didn't see them, but instead saw a few other things, like those really abundant black corals from the first dive that we did in this area.
I don't know what that is.
Maybe a piece of wood. That's my best guess, like a little plank of some kind. There's some nice looking boulders for you.
Yeah, we're full though.
Yeah, we're full.
I think so.
Can we zoom in on that sea star up there? Just the one straight ahead at the top. Yeah, those two. They're just cute. I mean, they're viciously attacking that really nice looking coral, but...
in hyper slow motion, but...
yeah. Look at those. That's great. Just wanted a quick zoom while we were on the way. Thank you. Wow.
Oh.
Wow.
Really neat little boulder crop.
Yeah. There's another fish in that one maybe, or 3 nudibranchs for sure, and then maybe a little fish at the top. Oh, 4 nudibranchs. There's one near the base.
Oh my.
And is that a fish? Could we snap on that? Yeah, so think a little juvenile rockfish. Just beautiful. That's amazing. So again, even this this coral that's being heavily predated on by nudibranchs and probably other things has a little juvenile rockfish nestled into the branches. Really beautiful to see. Just to document, when people wonder why we study corals and why we're, in some cases, setting up protected areas around them. You can imagine how vulnerable these would be to certain kinds of activities. So it's always really amazing to see them in real life.
And so as we were discussing with the biology, this is kind of exploration is a great example of what we can see with the geology as well. We're down in a bay, as I mentioned, in a place where people didn't try to map the geology back in the day. All the geologic contacts more or less ended at the shoreline. We know we had different kind of rocks on the nearest shores that are to our east and to our west, without really any idea of where the boundary between those two units are or is. And we've picked up a couple rocks here that will help us kind of settle that question. And it's something you can't do until you get down here, and until today we didn't even have the baseline information, which would be the mapping that would allow us to even get in here and explore. So it's really a neat example in a encapsulated way of like how you can make that push forward, collecting both the mapping data and then immediately getting down and getting the hands-on look and sampling to get some new information.
Yeah, it's pretty amazing to be able to do all of that, over the course of 24 hours, filling in all of those gaps in the map, looking at the species distribution predictions and picking kind of drawing an imaginary line and then going down and doing it. It's been pretty amazing. Wow.
Nice little, even kind of a spire.
Yeah. Some
almost.
Of these overhangs, it's hard to imagine what's keeping them up. Look at that.
Yeah, you can imagine that there was a boulder-sized chunk of rock in there that's dropped away, perhaps during some earthquake.
Right. Oh, look at this.
I mentioned the Queen Charlotte Fault has had 7 magnitude 7 earthquakes or greater in the last 100 years. The two most recent were in 2012 and 2013.
Oh, okay. Just a bit before I moved to the West Coast. Wow, look at that huge rockfish. So this has to be one of the older ones. Okay. Might even be gravid. I don't know. Definitely have to take a look and make a note of the video. It looks like it might be. And I know for rockfish, I'm pretty sure that the older they get, the more young that they have. So I think if you're kind of looking at a larger, older rockfish, they are the more reproductively active ones than the kind of mid-sized ones that you would think from other fish species. Some really big dead bases of some large primnoas here. Not a lot of, I think, kind of the remnants of larger species versus small ones recruiting. More of that sponge that we sampled earlier. I think those are a little patch of zoanthid anemones. Yep.
Oh, neat.
Yeah, definitely. So again, they're like the big solitary anemones we see, but they're connected at the base, so they're a colonial anemone. And they have sort of a long, thin, we call it a stolon in corals. I don't know what it's called for zoanthid anemones, but a piece of connective tissue that kind of encrusts over these rocks and connects all of those individual zooids.
Dud.
Wow. Look at that.
Wow.
Several different kinds of rockfish, some smaller looking individuals, so maybe juveniles of some of the larger species we've seen, like that one. Wow.
Really?
Huge.
Really interesting the way somebody stacked these rocks in here.
Uh-huh. Okay, a really curious rockfish as well. Somebody noted the black spots. I know some of them naturally have black spots on them, but some others maybe, some kind of scarring or damage. And that one looked like it might have been gravid, so gestating. Watch leads here. Maybe through how are we doing for pinpointing the scattering layer on these?
I don't know. I'll have a look and see if we can spot it.
Okay, stand by and we'll look into it.
See if I can spot it.
Okay, I wanted to double check before I talked about this, but I did think, and now I've just kind of confirmed that all species of the genus of rockfish that we're seeing, Sebastes, are viviparous. That means that they give birth to live young instead of laying eggs and leaving them to hatch. So they'll give birth to thousands or millions of tiny larvae. Very cool. Just want to make sure I get my fish facts straight before I mention them. Looks like that Hippasteria kind of is taking advantage of this
Looks like
really large primnoa, or possibly had something to do with it. This branch toppling over. Wow, that one's huge. But there's no shortage of primnoa growing here. These really large colonies. Another one of those sea cucumbers. Some smaller rockfish. What do we think about the Niskin? It's kind of hard to say. Maybe we do take one on the bottom.
This isn't a call I can make. I don't see anything that jumps out at me.
Okay, ROV, I will request a Niskin while we're here. Thank you. Get some really good eDNA data on a very unique and dense population of primnoa and the kinds of associated fauna that we see in the area.
At 314 meters depth. Sample ID EX2306-D11-13W.
Wow. I don't think I need to request a zoom. Let's take a look while it's still. An excellent opportunity. I've never seen one just kind of hanging out on the seafloor like this.
Oh, fantastic.
That is very cool. Yeah, did I see it change color a little bit? Yeah, because we've had a question asking if it was alive. Because yeah, I've never ever seen one that's just kind of hanging out like this on the sea floor, but I definitely see it moving now.
Mm-hmm.
Wow. Oh, this is an opportunity to get a nice zoom and imagery of this squid that almost never stay still like this. You only ever see them really quickly darting through the water column. And you can see all the little, I think they're chromatophores just pulsing. Look at those dots on the mantle and on the skin of this squid. You can see them kind of pulsing different colors. Just stunning. So other families sit on the bottom, but I'm glad Michael is.
That might be it.
Whoa,
and there he goes.
Wow.
That was neat.
So a gonadid squid of some kind. That's very cool. So also, we have Gonatopsis borealis. Gonatopsis borealis. Yep, I guess it is alive. There's a little bit of ink left behind as well. Wow.
There he is again.
Okay. So very capable of swimming very fast. Just taking a little break. Doesn't sound like any of the scientists in our science chat have seen that behavior before, of a gonadid squid just kind of resting on the sea floor. Wow, it just doesn't end with this dive. Look at all the all of the things that we've seen. It's just checking out a really nice view of the marine snow. We're kind of taking a look ahead. Just seeing if we could get to the bottom of this feature, but we're kind of pointed the wrong way. I'm happy to spend it looking at this feature. Wow, quite a few shrimp and arrowworms. Can we get the lasers back on? Thank you. Wow. Oh yeah, could we get a quick zoom on those again? We saw them before and I still don't know what they are. Would we have time to attempt a sample? I think so. I think we have C4. Okay. So I think, yeah. We've passed by these little unidentified sort of stacked discs. I think they could be eggs from something, but I really don't know. And it looks like there are a couple of brittle stars nearby that might get opportunistically sampled as well. I think we have to call these unidentified and maybe rare taxa. Yeah. Yeah, kind of little stacked discs. I see one in the jar. That's great, actually. We don't need all of them. Yeah. Perfect. Cool. Yeah.
Sample ID EX2306-D11-14B in suction sampler canister 4.
Wow, some really big Hippasteria sea stars. Thanks for that. Very excited to find out what those are. That's the dive. Whew. Wow, what a dive. This has been wild. For somebody that studies corals, this has been astounding. You know, we kind of started off the dive with that sort of sparse sightings of corals, and then at some point we just hit the right depth and we hit the right conditions, and we have found an incredible abundance of Primnoa pacifica, forests of them basically, some incredible imagery, incredible opportunity to ground truth this model that I was given access to. And yeah, I couldn't be happier. I think we'll be studying this dive for quite some time.
Yeah, and I think there was a nice contrast between the early in the dive, the steeper, cliffier parts, where there was certainly a contrast in what we saw once we got to the top, and so possibly that's information that would go into improving the models a little bit.
Mm-hmm. Yeah, definitely.
If slope is not already part of it, it's certainly a little piece that they can think about as going forward. So yeah, kind of neat. And we also just got some nice samples for some just geology ground truthing, and making a little progress with the seafloor geologic map. So all in all, a good day.
Yeah. I'd like to extend extra special thanks to the GFOE team, the ROV crew, and the video crew for the excellent sampling and imagery we were able to collect today. Thanks also to the scientists that tuned in on Labor Day, no less, to help us with the identifications and advise us on collecting. We always appreciate your observation and expertise, so thanks so much for joining us. I'll send a preemptive thanks to the lab crew, everyone that's going to be working in the wet lab a little bit later today than the past couple of days, going over and meticulously cataloging and processing these samples. And of course, NOAA Ocean Exploration and the Okeanos Explorer for the platform and for making this all come together and happen. So I'd give everyone a preview of what we were looking for tomorrow, but I think it's very, very weather dependent.
Yeah.
So you'll have to stay tuned to the social media and to our emails if you're on the shore-based science team for an update as we decide where to go. I think the weather's opened up a little bit back in the Gulf of Alaska, so we might be back on to our close to our original plan. So stay tuned for that information.
Yeah.
Thanks everyone for watching.
Yeah. So, all right. Yeah. Thanks everybody for watching, and tomorrow I'm interested in finding out where we go tomorrow too.
Yeah.
So I know we'll be back in the open sea. The question is, we're actually still dancing around some coming storms later in the week, and so we'll probably have a big meeting tonight and kind of work out ways forward.
Right. After the lab. After things are sorted, hopefully.
Right.
Or during. We'll see.
So till tomorrow, yeah. Thanks everybody for joining us, and we'll see you next time.
Permission to continue with the recovery?
Yes, you do.
Pilot, dive super.
Dive super, pilot.
Is the control room ready to continue with the recovery?
Control room's ready.
Sounds good. Let's go ahead and ascend up to surface 15 meters per minute, calling out every 10.
Roger.
Ascending. Four 0 meters, wire out.
T245.
Three 0 meters, wire out.
D232. All's well.
Two 0 meters, wire out.
D223.
One 0 meters, wire out.
D215. All's well.
All right, pilot, we have good eyes on, streaming directly aft.
Pilot has visual.
Good copy. Let's go ahead and lock in auto heading, inverse the ship.
Auto head in.
Okay, Deck, let's go ahead and recover Sirius. Okay, pilot, tugger lines are on. Please provide some mass thrust as Sirius comes up and over the side.
Roger, dive suit.
All right, pilot, that's looking good. Let's maintain.
Pilot's 40 back.
It's looking good, pilot. Let's maintain.
Backing 30%. Pilot 20%.
Good copy, pilot. Winch, let's take back local control. Deck A-frame.
Remote disabled.
All right, pilot off stick at this time.
Pilot off stick.
Go ahead and leave auto head engaged.
Auto head in till halfway down.
Super nice. It's like a daisy chain recovery machine.
Okay, pilot, let's disengage auto head at this time.
Auto head out.
Pilot off stick.
Thrust 0.
Okay, pilot, recovery line's clear, daisy chain's free. Okay, pilot. Lift line's going tight. Let's go ahead and lock in auto heading, inverse the ship, start backing towards crane.
Auto head inverse, backing.
Give us a slight bump to surface, pilot.
Bump up.
Let's ease your back a little, pilot.
Easing.
All right, pilot, you can pick up speed back again.
Picking up speed, bump to surface.
Good copy, straight back.
Straight back. 40%.
Looking good, pilot. Let's maintain.
Maintaining. Bump to surface. Bump.
Good copy, pilot. Keep coming straight back.
Coming straight back, about 60%. Bumps up.
Good copy, pilot. Keep coming back, almost under the crane.
Back in 60.
Give us a little port lap.
Come in port.
Copy, thank you. Looking good, pilot. Just port now.
Port only.
And let's ease on that port.
Easing.
And you're under the crane.
Backing.
Let's give us a small twist to starboard.
Twist to starboard.
And pilot off stick.
Pilot's off stick.
Pilot secure power.
Deck, E-stop. Winch, knife switch.
Knife switch.
Voltage read 0.
Vehicle's cold. Pilot Dive Super.
Dive Super Pilot.
Trolley straps are going on. Feel free to wrap up in the control room and come on down for post dives.
Roger, Dive Super.
Good recovery. Winch, Dive Super.
Go for winch.
Feel free to secure the HPU at this time. Come on up, help with post dives, and please hold off on turning on the fans until after we're done.
Good copy all.
Bridge, Dive Super.
Yes, go ahead.
I'm aware that you now have control. If I could just ask that we maintain a steady ride as we do post dives.
Sounds good, thank you.
Thank you very much. We'll call up when we're off the ladder. If there are any logs or anything that you need to avoid, just feel free to call down, let us know, and it shouldn't be an issue to move a little bit to get out of their way.
Perfect, thank you.
Thank you, Bridge.